in soybean production. Soybean is generally suffered with many diseases. Anthracnose is one of them. It is very main disease to create a huge loss in production of soybean. Coledotrachum species is responsible for the anthracnose disease in the Indian continent. Other species also there, but it is a major reason for crop loss in Indian continent, Indian region. There are several methods for controlling the disease, like cultural, biological, chemical, but all have their own limitations. So, to overcome these problems, we have to move on a new method, that is uh, creating resistance cultivar. And parallelly, that creating resistant cultivar is have their own benefits. We have Jiva's panel consists about 300 lines subjected to screening anthracnose disease. Here we use a new method, in vitro detached pod inoculation method. Earlier, uh, other methods also there, that are field screening. These methods have their own limitations, like they have to suffer with the environmental conditions. But in our in vitro method, we are doing all the working in a controlled environmental condition. So they are giving much more accurate results. Me- Methodology. We uh, we cultured pathogen. We isolate and cultured pathogen in laboratory condition. Later we sampled pod. We are looking for resistance at pod part of the plant, because it is very much sensitive for this anthracnose disease, other rather than other parts like leaves, stem, and other. First of all, we prepare pathogen culture samples the pod from the field at particular stage, like 20 to 25 days after flowering, which is perfect stage for this purpose, because other stages results are not liable to calculate the resistance potential, because over mature pods are not good, as well as immature pods are not good for these experiment. Later on, they are soaked in uh, suspension that is followed by keeping that pods in BOD incubator, which is maintained a temperature ranging from 25 to 20 degrees centigrade, and humidity from 85 to 90%. And observations are recorded at three specific time points, that are 48 hours, 72 hours, and 96 hours. We also grown a check GS9560 parallelly to check the susceptibility at what time point it is giving susceptible. That time point we will use to evaluate the resistance potential of our Jivas panel genotypes. Readings are taken in percentage form, means percent area of tissue that is covered by the disease. Results for all three time points, there are different range of disease severity. That is, for 48 hour it is 0% to 30.27%. For 72 hour it is 4.45% to 43.34%, and as well as for 96 hour it is 9.86% to 83.37%. And the average of these disease severity rates, 5.33%, 15.56%, and 29.63% respectively for these time points. By the looking into these results, we identify some genotypes as resistance. They are like PUSA 37, EC33876. Some moderately resistance like EC5468882, and parallelly some susceptibles like EC389163. By these results, we reached on the result that we don't find any immune as well as any highly susceptible line. Most of them were resistance, Few are moderately susceptible, and one is resistance. Sorry, moderately resistance, or which is we considered according to 96 hour of time point, because on that time point we are getting our check susceptible. Figure 2 is showing frequency distribution of three time points. Like we for 48 hour it is maximum at 5, for 72 hour it is somewhat maximum at 15, and for 96 hour it is maximum between the 15 to 30. This is a descriptive analysis of our given data. We have mean of different one, their average, their maximum and minimum value. Other figure is showing auto correlation between three time points. Auto correlation means correlation between three time point of a single variable. By this table, we can see how many we are getting immune, highly resistance 1, resistance 2 to 8, moderately susceptible 45, susceptible 15, highly susceptible 0. Next one. This picture is showing the ports that are be taken. One is resistance and other one is showing susceptible. By looking, we can see that the susceptible ones are totally covered with disease. They are covered by pathogen growth. Conclusion, like we identified different genotypes in different for different anthracnose potential for resistance. Further utilization of our findings. As uh, as it is a running project, we have some results which will further compare with earlier results we got in last year season. They compile them, and they go for genetic screening, genetic evaluation of to mm, find out the real factor for resistance behind the real factor behind these resistance, and they will they will incorporate these resistant factor into new varieties by different breeding programs and generate new cultivars. It is benefited for farmers also, because it creates a less dependency for farmers on the pesticide application, because currently they are using a peak of pesticides for getting more and more production. High production productivity is also a major target for these, and cost of cultivation is also lower. And earlier I mentioned it is a eco-friendly method as well. Thank you.
Uh, yes, Mr. Pavan, it was a good presentation. But as you are saying that no variety was very highly tolerant or very susceptible to that a particular anthracnose, isn't it?
Yes, ma'am. Could you repeat, ma'am, once?
Yeah, you were saying that none of the variety was very susceptible or tolerant to the anthracnose.
Uh, I told there is no immune, but we are getting resistant variety. That resistant variety we will use for further cross with other ones. Or, ma'am, we are like in this institute they are trying again and again for getting immune, but generally we don't are getting that.
So, how much yield reduction will be there because of this particular anthracnose in soybean?
And generally, it in my region I observed it's like 60 to 70% reduction we are getting in
Yield
overall yield.
reduction, yield reduction.
60 to 70% when it is at peak.
But I think that is very higher end of that one. 60 to 70% is very higher end, no? So what is the major, organ that is mostly affected by this anthracnose?
Sorry, ma'am.
Part of the crop, which part?
It is generally like nearby me. Like I am living in Indore. I'm looking this. I'm getting this disease, and nearby Khargone and other districts. So I also seen. I heard from my some friends.
So it's only leaf, isn't it? Mostly leaf
No, ma'am.
anthracnose spot.
Actually,
Hmm.
ma'am, it is affecting all the upper ground parts like stem, pod, leaf. But it's a severe for pod that leads a reduction of
Yes,
disease, ma'am.
when it comes to the pod, then only I think severe reduction of 60% may be possible. Otherwise, leaf and all, when it is at a later stage,
That
it may not be so
that
harmful.
what tolerance for that stage.
Other than this anthracnose, what are the other diseases which are having a major setback in yield in your
Other
area?
than anthracnose, ma'am, like RAB, Rhizoctonia aerial blight, bacterial pustules, and little bit mosaic also.
Okay then.
Pavanji, can you please tell me the limitations of solely if you are relying on resistance breeding to combat this anthracnose in soybean? So, and how can you how can IPM approaches complement genetic resistance strategies for sustainable disease management?
Like if only we are relying on it, it will take time, ma'am, time consuming as well. Because like for these same data, last year they completed one experiment, and again this year I'm doing for that, and still they will go for molecular identification of that particular factors. Then they will go for breeding purpose. So it's a time taking process, ma'am. Other than
You
that,
have you have many limitations, like you'll find genetic variability, right? Resistance breakdown, narrow genetic
also
base,
some
and
things, ma'am,
the
we are
IPM
getting
approaches,
phenotypically.
they can complement genetic resistance strategies for sustainable disease management, right? By enhancing plant health, minimizing reducing the chemical inputs, adapting to changing conditions and all. Okay. Any other questions from the audience?
यदि ऑडियंस का कोई क्वेश्चन नहीं है तो नेक्स्ट के लिए मैडम आप प्लीज बोलिए.
Next presenter, please, please start the
Dr. Ashok,
presentation.
are you there? Ashok, Dr. Ashok P.
Sir, good afternoon, sir. Shall I share?
Yes, sir. Yes, sir.
Please allow, sir, for sharing. Sir, please permit me for sharing.
रोहित जी को होस्ट बनाइए आप इन्हें अशोक का शर्ट को. हाँ सर, आप को होस्ट है, आप परमिट आप हैं, आप स्क्रीन शेयर कर सकते हैं. आप स्क्रीन शेयर कर सकते हैं सर. परमिट की कोई जरूरत नहीं है उसमें.
Not permitting, sir. Okay, sir. Okay. Shall I visible, sir?
Yes, Daksha.
Okay, sir. Good afternoon, sir. Respected chairman, co-chairman, dear all audience. I would like to present here millets is a smart food first crop. I am working as a professor and head. Earlier, I worked as a millets as a senior scientist and head in the research and extension work. I would like to share the some millets work is as a smart food for 21st century. You know the year 2023 is a celebrated international years of millets because of it is healthy food, gluten free, dietary fiber is the main kind of. This is also very celebrated all over the world. India is a international celebrate India in a millets year. And among the millets, no further, these millets are climate resilient crops because of around more than growing 131 countries. Among that, during 3000 BC, major millets, minor millets, among pearl millet, sorghum, finger millet, foxtail millet, kodo millet, little millet, proson, barnyard. Another you can go for the these brown top millet. Recently, I have added two millets, teff and ponio millet. These are also very good for the scope of these millet crop. Barley out to gluten-free traditional practices is also growing very well response in the country because of more than 67% is our India rainfed agriculture. This crop is highly suitable and also very nutritious food of the cereal among the peoples. These crop is you can show for the these are sorghum bicolora. These are the millets of mission in sorghum crop. In finger millet, this also you can go for the rich in nutritive value cereals compared to other crop.
Start
In
moving.
barrier millet, then
Slides
proso
are
millet. This
not
is also you
moving,
can look
sir.
at
Please
proso
note.
millet.
Sir, excuse me, sir, slides are not moving.
Oh, now is moving, madam.
Yeah. It's going ahead. Go ahead, please.
Okay, thank you, madam. Thank you. Already is a barnyard millets. This also we can very good for proso millet. You can throw out here. You can growing this very good for the fodder crop also. This is in foxtail millet. Is because of it compared to other nutritious food. This is also very excellent food in our India. This is also major growing in Chhattisgarh, Karnataka, some Andhra Pradesh. This is also major in dryland states. Almost this is also can topmost producing yield. This is also little millet. It's very good for the you can fodder qualities also as well as human as well as also animals. Another this is also kodo millet because of yield potential is very less because also very good for nutritious food of the in kodo millet. Among that, this also you can brown top millet. This also you can very good for the more tillering and also here is more yield potential. Recently introduced in our India ergo street cap. This is also the staple food of Ethiopia. This is also very good for you can millet crop. Our CFTR Mysore and IMR Hyderabad is developing many varieties. And now one of that I am also working as a ADAC project. This also TEF is a nutritious as well as a you can good crop without processing. You can directly you can consume these crop. This is also Ponia crop, especially the Poaceae grassy family, but is also you can some African countries is commonly growing up this millet crop. This is also another additional you can new millet crop Sikia. I can go for Sikia. This is also very important. You can go for the these also seeds are very. This is also you can one of the weed, but this also you can millets is as a can seeds is used as a consuming in millets in African countries. Or whatever you can go for these millets are you can known for dryland crop, high tolerant, water adaptability, and then also you can long duration. Another you can brown top and is a nutri rich. This also you can pet name is also you can named as a millet crop. These also you can different names of you can local languages. The present global scenario in Africa and India is like also leading in area production and productivity. But even though some parts of Asian countries also you can growing is very well in among the more than 1 lakh ton millets of production in all over the world. These are also global millets. Area and production you can go for nowadays is productivity is decline because of you can go using own seeds, but you have to achieve the last year 20 years. That also increases the production of the crop. That's very important. This is also world area production millets crop. Along the you can mean of the production area. This also you can decrease 20 million hectares because of you have to diverted crop diversification. Another you can go for these crops are very small seeded harvesting is another issue is some we can research studies is shown. Among that is a green revolution period. Also millets is very less, but you can after productivity and production after 1970 in India and after 1966 This also you can only for increases the millets production. Among these millets are jawar, bajra, ragi, small millets is also very good for production in percentage of area is increase among the different state wise. These are also millets in more than 56% is can growing, but you have to productivity is very less. They also you can go for the thing for research and the extension gap is another important issues of millets productions. This is also consumption. Almost India is the major consumption. Nigeria and China I can go for the compared to other countries. Another this also now we have to now biofortified millets varieties also introduced based on zinc and iron content. Our prime minister is we can go for the main focus of the we can go for the nutrients wherever we can imbalance of nutrients. There's also many research we have to is major millets is also used as a biofortified millet varieties. These also can distribution small millets in India all over the India. These are some major states, especially you can go for already discussed Madhya Pradesh, Maharashtra, Chhattisgarh, Punjab, Rajasthan. These are some major states is growing. Another you can go for Gujarat and Haryana. Some parts of the Bazar is a major growing, major millets of the crop. These are some stage-wide distribution of the state among the foxtail millet, pearl millet, and sorghum and minor millets. These are the percentage of contribution of India. Is also major. You can go for the kodum millet and little millet is higher than the foxtail and proso millet of the. These also because of very short duration and also you can very good nutritious for consumption of the peoples. Another important issues. These are the annual growth rate. You can go for the annual growth rate among the area production. Sorghum and pearl millet, you can finger millet is very less production, but even though small millets is higher production and productivity of the compared to major millets. These are per capita monthly consumption. Sorghum is a common known for C4 plant, drought resistant, you can measure food staple. Even though sorghum and pearl millet you have to followed by finger millet is also very good for the consumption of the as a rural as also urban. Nowadays as a diabetes patient major consumption of the these millet crop. You know the things already told international millets years of 2023 because of this is second zero hunger, cold health, also decent work, this is also climate action. Another important you can go for the around 2030 agenda is that you can go for the water based on 2030's agenda. We celebrated international years of millets, sustainable development and sustainable development goals. This is called SDGs is another important issues of the code. Millets is called superfood. You know the for heart disease, cancer, arthritis, strokes, respiratory, another healthy issues. This is also because of high volume minerals, and antioxidants. But nowadays is many research issues you can go for many you can production of value added products in market also Reliance more many market is online is available due to you can superfood. Millets have is high low glycemic index. You have to good for the in the sometime diabetes patients. This also you can already know the for not only health is also climate resilient crop because of nowadays is global warming, climate changes, international is many debates. In among that, these millets crops are very grow for marginal small farmer, tolerant, drought tolerant, less water requirement, less nutrient requirement. Another is photo insensitive. Another important issues is also you can go for the millet crops. You know the special feature already discussed. Many you can millets are you can very old and know for mid in food grains. Another you can go for these poor countries, poor families. Definitely this is also where you can go for the food of the we can measure for the agriculture is a major consumption. This is also future research trust areas you can based on crop wise sorghum, pearl millet. You can small millets is another important you can need for the can production hybrid technology. Improve the bioproduction. That's definitely you can go for. Not only you can satisfied in human in animals you have to go for because of little millet is a more straw production. You have to compare to other millets in finger millet and foxtail millet. Another you can go for development of genomic research. Another you can trust area. Water and nutrient use efficiency. Another important you have studies for the. Also very less you can go for the pest and disease incidents. There's also global seed market is another demand. Definitely India specific nations is also increased food demand. This based on the seed. This is also one more protein and fatty foods are very important for the consumption of these millets. This is also world area production productivity. Among these countries, seed production requirement is very demand for the high yielding potential of finger millet, proso millet, and foxtail millets. This also nowadays is you can go for the India major millet export. Among the market intelligence report, global market. This also you can 2023 you can go more demand for the according to CASR 4.6% is increases during 2019 to 2027. That's very important of the in Asian specific. Among the total value of the millets in India are very less per I can in need for I can per capita is in need for more, but the production is very less among the based on the domestic consumption, value addition, value in exports is very, very important for the total value of the millets in India. These are some percentage of increase of production millets. Among those 2020, 2030 and 40, 5%, 10%, these are because of, these are very climate resilient, very smart crop. This also another you can go for very super food. This is also very demand of these millet crop. These are healthy benefits. This already discussed. This also you can go for healthy minutes of this also commonly you can know the very well. Another you can go for national nutritional composition is very, very important to rice and wheat. Nowadays is wheat is high gluten content. Rice is we can go for the rich and we can go for the carbohydrates. That's we can go for the comparison of these all millets. Very good for the, we can go for the, these also you can the nutritious composition. Amaranth seeds, quinoa, buckwheat. These also cereals is also more competition, proteins, so diabetic fiber, and we can go for the, these also riboflavin and mucin. Folic acid is another important millet crops. Our Prime Minister, this is what we can go for international years of millets celebrated. India is the honor to be at the more frequent propagation of the millets. Millets consumption, further nutritious, food security and welfare of farmers. Our Prime Minister is they can go for the more canvas of the all over the world. Millets is we can introduce very good for the superfood. We can go for move on value-added products. Our IMR, many KVK extension works. There are also many we can processing technology. And so popping, baking, because of value add you can process millet, paco millet is also millet flakes. Another you can go for KVK because many population of these value added products. You can go for the among the you can go for as a use as also you can go for the some chiroti rava or finger millet. This is also very good for cost of cultivation. Very also very less. You can also benefit cost ratio is very high for the these consumption. This also you can go for the in SSG group, self-help group. They can go for the we can make a brand. Finger millet vermicell. Vermicell is another important use as a wheat and barley vermicell is common much demand is that is our KVK extension work is millet the value added products. Another important things of these things. You can many products also you can based on the consumption. Another you can go for the you can SSG groups and you can go for the Narega activities also you can go for the very good for consumption. These also you can go for the value added you can tastiness, value added production based on the upama preparation. Upama is also major things. Our woman is also making the millet made because of nowadays is very good for food. That's also you can self-employment created the self-employment for the value added products. That's why nowadays is strategy policies intervention is nowadays government of India many nutri cereals as a used as strategy policy. Lower yield gap. This also we can go for the season wise. That's why is also strategy is a measure we can make for the works. This is also important constants of nowadays. Lack of knowledge, lack of research, definitely. Because of these also you can neglecting crop. But now many research institutes, IMR is developed many yielding varieties last 5 years. Nowadays is more you can farmers is you can awareness for the today you can grow for the yielding varieties. Another you can market network is another important issues. After international millets is celebrated, many government agency, state government agency can more canvas. These also you can these also value added products. Nowadays is you can go for the
I think
value added products directly you can available.
You are nearing the time, I think already.
Okay, madam. Okay, madam. These are some major constants you can agronomic practices because I tolerate high water products. These are some major important things. Our US Dharwad released many varieties in foxtail millet, lentil millet because high yielding potential more than 20 to 22 quintals in production of the lentil millet, barnyard millet and proso millet. In agronomy practices, you can go for this very good for agronomy practices, timely sowing, optimum nutrient management. Definitely can go for achieve the more yield. Another important. This also you can very important for data of sowing, optimum nutrient intercropping. Intercropping is additional income of the main crop. Irrigation management because sometimes little millet is growing in summer. You can go for 1 or 2, 3 productive irrigation. Definitely achieve the throughout the year in the summer month. This is also very important. This is also less water requirement. You can compare to other crops, sugarcane. You have to go for other cereals and pulses. You can go for millets is very less water requirement. This also you can show for higher water usage since it is millet crop. That's why nowadays is we have to rob out ourselves by switching to organics. But nowadays is organic farming is more focus. That in organic focus, you can go for the production of the millets is very good for you can also you can growing of the millets is in organic farmers. This is also very important aspect. Can finally I conclude my topic. Eat millet, stay healthy for food and nutrition security. Definitely we have to we can more focus in whatever you can go for the agenda of 2030, millets is superfood. Thank you, sir.
Sir, you have focused on good presentation.
Please, madam. Yes.
But I have one doubt is that like Karnataka and some other places who have their staple food as one of the millet, the area is flourishing in that region.
Yes, madam.
But until and unless we replace this rice or other wheat and so, these millets generally will not get the required name or the area. And
Definitely,
even
madam. But nowadays,
organic also we are saying, but with respect to farmers and all, they are not showing much interest in the millet increase in the millet area. So it's only pertinent to the what is the theoretical or this thing, but we are not seeing practically either increase in area or demand from the farmers to go ahead with these crops.
definitely. But madam, of the government of Karnataka, government of India, many Krushi Vigyana Kendras. We have many frontline demonstrations, seed production because of our. I don't know other states. Government of Karnataka is they can go for can incentive amount. 1 hectare is 10,000 rupees for growing all millets. But nowadays is you can go for Chhattisgarh and you can say the imported and exported is also going on. Collaborated CFTRI, many production evaluated. Our ICR Institute, IMR Hyderabad. Many you can go for self-help groups and also human youths. Many making in online offline trainings, preparation of evaluated. But is healthy issues, madam. Future is definitely can scope for millets.
Okay, sir. Okay. And something you have focused on, especially that Digitaria sanguinalis. I hope
Yes,
as a weed expert also, the seed is very, very tiny enough, very small. I just have a
yes,
doubt
it's
like
very small,
how
madam.
it will
When
be
I yes, yes.
mainstreamed into the food line.
Yes, madam. When I went in ICRISAT, one of the scientists is small part studies. Because I gone for reviews. Africa is a commonly consumed whatever you can wasteland as a millet. That is I wondered. Just I add the information here, madam.
Yeah. That needs something still more. What is a research study to be focused on that, sir?
Definitely. TEF is another important, madam. TEF is you can whatever Ethiopians.
Yeah. Yes, sir. I have seen TEF and Konya are okay,
Very
but
gluten free, very good nutritious because very.
yeah, this Digitaria only I had a little doubt. Still studies need to be focused on that, I hope.
Definitely. Yes, madam. What you were saying, yes, exactly, madam. Because the agronomist, also millets agronomist, definitely plan for conduct this research, madam. Already submitted on state level, our state government in ADAC project, definitely plan for in this area. You can go for research, is future research and research.
Sir, it was a vast presentation done in a small amount. Good presentation.
Thank you, madam. Thank you very much. Thank you. Thank you, chairman and co-chairman. Thank you.
A very good presentation, sir. Sir, can you please suggest some steps, sir, that can be taken to integrate millets into mainstream food system, sir?
Yes, madam. Because of already this is also TEF is very short duration, 70 to 80 days. But even the harvesting is another important issue. And you can go for the very small seeded. You can now no need for the you can go for the whatever you can go for the in processing. Directly you can go for whatever you can very small seeded. You can mix it with sand and also insert material. But our CFTR is conducted research. Our US Darod myself only conducted last 2 years. But it's very difficult for you can in harvesting and we can go for the issue. But nowadays is you can screen for future research, madam. Definitely.
Okay, thank you.
Okay, madam. Thank you.
I hope the next person could be ready in sharing the screen.
Yes, ma'am. Yes, ma'am.
And you can share the screen.
Okay. Am I visible, ma'am?
Yeah, your screen is visible, please. You can go ahead.
Okay, thank you, ma'am. Good afternoon. I am going to deliver my presentation in this international conference on recent advances in agriculture for ATMANIRBHAR Bharat. Irrigation and nitrogen scheduling through drip fertigation in terms of growth and yield of maize. So let us start with the introduction. The maize crop is the world's most leading cereal crop, also due to its great genetic yield potential.
Madam, that the slides are not moving. Just, I think it needs a little time. You just try to share the next slide, please.
It is moving now.
We are in the first, I think, first
Ma'am,
slide, title slide, we are there.
Still not moving. Am I audible, ma'am?
Yeah, you are audible, not an issue. Yeah, now the slides are moving, please. You can go ahead.
Okay, ma'am, is it on crop now? Moving.
Yeah, the title slide is visible, madam. Title slide is there. It has stuck up there.
Moving now.
Yeah, you are audible, madam, but the slide has stuck up at the title slide.
Ma'am, it's stuck. I don't know why, because I am moving it, but
now it has started. Just you can go ahead, please.
okay, ma'am, it now.
Yeah, you are now at introduction slide. Please go ahead in a faster pace.
Okay, ma'am. Thank you, ma'am. So after the rice and wheat, maize is the most significant crop in the India that accounts for nearly cultivated in the variety of seasons. Like you can take maize in summer as kharif and a rabi crop as well. So we also included the drip fertigation in the study. And drip fertigation is basically the application of water and fertilizer as well as fertilizer, and ultimately enhancing the crop productivity. Three treatment of irrigation and 6 treatment of nitrogen were taken. In the main plot, it was a split plot design. So in that plot, the irrigation treatments were taken as 100% and evaporation and 80% and 60%. And in the subplots, the nitrogen treatments were allotted, and then another 5 treatments of nitrogen were taken, different scheduling in as basal and fertigation. So objective of my study was to optimize the time of nitrogen on growth and yield in maize. So the key findings of my study was uh, when I the plant height that is 195.2 centimeter was highest that was recorded from the treatment applied with 100% pan evaporation irrigation level, 80% pan evaporation, but it was significant pan level. And in fertigation treatments, it was found that the treatment applied with 100% recommended dose of nitrogen maximum plant height, that is 196.7 centimeter, F2, that is 25% RDN application as basal, along with 75% RDN to fertigation and fertigation. And this treatment F4 is significantly superior over the treatment F0 that is taken as the conventional practice. But
The voice is breaking and it is not at all clear.
RDN is applied as
Don't know how it
plus
is for others, but the voice is
67%
continuously
applied
breaking.
in two
Yes,
split
ma'am, the
doses.
voice is breaking in between. It's not clear.
The slide has stuck up at the crop introduction itself.
Similar trends were observed in dry matter accumulation, maximum dry matter accumulation in gram per plant was now issue. Actually, the previous candidate was clearly audible.
Ma'am, I think we can move with the next participant, right?
Yeah, because I just don't know.
Yeah,
It has been broken and not clear. Let her
there might be
come.
network issue. So,
Yeah.
Dr. Dimple Compose, are you there?
कंबोज का करवा दो, कंबोज का.
जी, Dr. Dimple Compose, are you there?
Yes, sir.
We can proceed with you.
हाँ कंबोज जी, आप स्टार्ट करिए.
Sir, I am audible, sir.
Yeah,
Yes, sir.
ऑडिबल हैं आप, ऑडिबल हैं.
And screen is visible, sir.
हाँ जी, हाँ जी.
Good evening, everyone. Today I am going to present my presentation on effect of different timing and level of nitrogen application on fruit yield of chili in alluvial plains of northern India. Introduction. Chili is the one of the most significant vegetable crop belong to the family Solanaceae, which is used for spices, pungency, taste, and culinary purposes. And it is mostly grown for their aromatic fruit and rich source of vitamin A, B, C, and it is prominent for nutrition value in addition to the flavoring agent in food-based industry. Nitrogen is the primary nutrient element which is a structural component of protein, play an important role in the plant physiology, improving the growth, yield, and development of the chili. The widespread use of the nitrogen fertilizer and the limited use of the FIM in modern agriculture result in the loss of soil organic carbon and multi-nutrient deficiency. India consumes 16% of the nitrogen fertilizer globally, so higher fertilizer consumption in developing country due to the knowledge gap between the farmer and scientist and availability of the highly subsidized nitrogen fertilizer. So that the nutrient management technique like split application of nitrogen can increase the fruit yield and total nitrogen uptake, which help in reducing the nitrogen losses and improve the nutrient use efficiency along with the fruit yield. And the integrated nutrient management, that is the integration of the organic manure, that is farmyard manure, with the nitrogen fertilizer, help to maintain the optimal soil fertility and adequate supply to maintain the desirable fruit productivity. Objective: to assess the productivity of chili hybrid under different nitrogen management practices. And the location is Research Farm Department of Soil Science, PAU Ludhiana. And the cultivar use is CH27. The statistical analysis was done with the CPCS1 software using LST less than 0.05. Material and methodology. There are 2 main plots and the 13 subplots. And in main plot, there is without FIM and with FIM, that is 25 tons per hectare, that is recommended as per package of practice of the Punjab. And in subplot, there are 13 treatments. The first one is control without nitrogen, and T2, 3, 4, 5 is 75 kg nitrogen per hectare in 2, 3, 4, 5 splits. And the T6, 7, 8, 9 is 113 kg nitrogen, 2, 3, 4, 5 split. And T10, 11, 12, 13 is the 2, 3, 4, 5 split 150 kg nitrogen per hectare. Now the phenological stages. The nursery raising on 30 November and the transplanting done was 19 April, and the split dose were applied according to first at the transplanting, then at every picking. And the number of first flowering is done on 7th May, and the fruiting is done along with the split nitrogen doses. And at the later stages, the physiological maturity come at the last in 31st of August when the crop fully get dried. Initial physiochemical properties of the experimental soil. The bulk of the particle density 2.61 and the texture class is sandy loam, and the pH is 7.44, that is alkaline, and EC is 0.23, that is normal interaction, and the soil organic carbon is low and available nitrogen is also low, and potassium and phosphor, potassium and all the micronutrients are in the optimum amount. In result, effect of different timing and level of nitrogen on number of the fruit per plant. The data presented in the table show that farmyard manure show a significant increase in fruit yield as compared to the without a farmyard manure plot, and it was further noticed that the different timing and level of nitrogen significant effect on number of fruit per plant. The higher number of the fruit per plant were found in farmyard manure plot, that is 252 as compared to the without FIM plot. And the higher fruit yield was obtained in the 113 kg nitrogen per hectare in the 4 equal split. The interaction effect of farmyard manure and nitrogen was observed to significant, and the F25TA treatment, that is 329.9 quintal number of the fruit per plant, having 9.23% higher fruit yield than F0T12 treatment, that is 150 kg nitrogen 4 equal without FIM, and the number of fruit have the ability to plant to reproduce the flower and fruit production, and there is a positive correlation between the number of fruit per plant and the fruit yield. So the incorporation of the nitrogen fertilizer result in higher fruit yield up to 113 kg nitrogen per hectare, but after 150 kg nitrogen there is increase in vegetative growth, so the 113 kg nitrogen 4 equal split have the higher fruit yield, so that number of fruit per plant is also more in that. Effect of different timing and level of nitrogen on fruit yield at different picking of chili. In this, there are 5 picking, and the second picking has the higher fruit yield, then first picking, then third picking, then fourth, and the last 5th picking, there is physiological maturity. In this, the farmyard manure plots have the higher fruit yield as compared to the without farmyard manure in the first, second, third, fourth, and 5th picking. And 113 and in the first picking, the 150 kg nitrogen had more fruit yield as compared to the other plot. But in second picking, 113 kg nitrogen had more in 2 split because the nitrogen fertilizer applied in 2 split. But after later stages, third and fourth picking, the 113 kg nitrogen per hectare in fourth split and 130 kg nitrogen in 5 split had the more fruit yield. But at the 5th picking, the 113 kg nitrogen had the more fruit yield as compared to the other treatment. So if we overall compare the effect of the total fruit yield of the chili, then the farmyard manure plot have the higher fruit yield as compared to the without farmyard manure plot. But then the 100 TA treatment, that is 113 kg nitrogen in 4 split dose integrated with 25 ton per hectare farmyard manure resulted higher fruit yield as compared to the other treatment and other without farmyard manure plot. And the interaction was found to be the significant in fruit yield at total fruit yield of the chili. So that there are significant interaction, and the higher fruit yield in the F25 TA treatment, that is 134 equal split, that is more than the as compared to 150 kg nitrogen. And in this result, it was also found that 75, 113, and 150 kg nitrogen, 4 split is best than third, 2, and 5th split doses. Effect of different timing and level of nitrogen, total nitrogen uptake, and agronomy efficiency of the chili. In this, 150 kg nitrogen per hectare have the higher, in 4 split have the higher total nitrogen, and farmyard manure plot have the higher total uptake as compared to without farmyard manure plot. But in agronomy efficiency, 113 kg nitrogen have the more fruit yield, and the farmyard manure plot have the higher fruit yield as compared to the without farmyard manure plot. This is due to the agronomy efficiency increase in fruit yield per unit of the nitrogen fertilizer applied. In the last, in conclusion, higher fruit yield was attained in 113 kg nitrogen 4 equal split integrated with the farmyard manure 25 ton per hectare, which perform 45.8% higher fruit yield as compared to the recommended dose of fertilizer, that is 75 per hectare in 2 equal split, as compared to BAU practice crop in Punjab. The more number of fruit per plant nitrogen uptake within each level, split 3 and 5 with FIM plot, and the integrated application of the FIM with nitrogen improve the nitrogen along with the fruit yield as compared to sole application of nitrogen fertilizer. Thank you.
Yeah, Dimple, I think आपका पूरा टाइम सोइंग और हार्वेस्टिंग में ही हो गया होगा 78 प्लॉट्स करने में.
Ma'am, actually there are 5 split dose, and we can check the physiological maturity as well as the increasing the fruit yield at a later third and fourth picking.
Yeah,
as compared to the recommended dose.
Even you told that 113 kgs is better than the recommended dose of fertilizer. It means almost of 30 to 40% higher than the recommended dose. Even with the FIM has given higher yield. What do you understand by this?
Ma'am, actually we have optimized the dose in the package of practice. There is a recommendation of a variety. There is a nitrogen recommendation of variety, but CH27 is a hybrid. It is more responsive to nitrogen fertilizer. That's why we can optimize the 113 kg nitrogen, and in 4 equal split. Due to the higher fruit productivity of the hybrid as compared to the variety, we can optimize the 113 kg nitrogen.
You done the optimization, because with the optimization only you can say that 113 is the optimum dose. ऑप्टिमाइजेशन कर्व किया है क्या?
यस मैम।
प्रोडक्शन फंक्शन का कर्व किया है?
मैम फ्रूट यील्ड को मैम अपटेक के साथ वो कंपेयर करके मैम वो कोरिलेशन ग्राफ बनाया था अभी, जिसमें काफी वो प्रोडक्टिविटी फ्रूट प्रोडक्टिविटी इंक्रीज हो रही है और अपटेक भी इंक्रीज हो रही है।
Yeah,
न्यूटन यूज एफिशिएंसी मैम 75 से 100 की बेटर है मैम।
when you recommend to the farmer, optimization should be done क्योंकि this requires huge fertilizer. Without optimization, optimum yield कहाँ पर आ रहा है वो देखे बिना we cannot it cannot go to the farmer.
Actually, ma'am, the variety is fruit yield up to 110 quintal per hectare. But if we compare with the hybrid, its yield is 240 quintal per hectare. It is highly responsive, highly productivity as compared to variety. So it need more nitrogen fertilizer. And it's and at later stages, third and fourth picking, there is a problem. If we compare with the two split dose, it's for transplanting and first picking, and there are only 3 picking. But if we optimize the dose in 113 kg nitrogen 4 split, there are 5 split dose, and the physiological maturity come after the fifth picking.
And आपका FIM भी इतना ज्यादा है 25 tons. Whether it is available in that huge dose, availability to the farmer 25 tons per hectare.
Ma'am, without the farmer manipulator plot, ma'am, then it's 150 kg nitrogen in 4 split recommendation.
But you cannot go. But some alternate source should also be seen क्योंकि FIM का मिलना भी बहुत कम पड़ रहा है without a population and all with the farmers. Animal population कम पड़ रहा है ना all over India. There is no maintenance of cattle by the farmer.
Okay.
Okay. Okay, but I think it is a very huge study with a more number of plots and much work. Very huge work you have done.
Yes, ma'am.
Okay, but have some idea on that optimization and all. Okay.
Thank you.
अब इसके बाद में गंगाधर दुगाली है ना रोहित?
नो सर, सारावंत हिरला।
अच्छा अच्छा, करो मैडम, प्रेजेंट करो, फिर पोस्टर सेशन भी कराने हैं, थोड़ा सा शॉर्ट ही करके चलो अब
जल्दी। यस सर।
पोस्टर सेशन अभी पूरा बचा हुआ है,
You should be little
फिर कल का समय वैलिडिटी में और उसमें चला जाएगा।
15 minutes means it is very slow.
जी जी मैडम।
Am I audible, sir?
हाँ सुन पा रहे हैं आपको, आप कंटिन्यू करिए मैडम।
Okay, sir. Good afternoon, everyone. Myself, Shravanti Ila, PhD scholar from Professor Jaishankar, Telangana State Agriculture University, Department of Entomology. My topic is morphometry of hemocytes from healthy and Beauveria bassiana treated 15-star larvae of Pyrus brassicae, Lepidoptera peridae. Then coming to the introduction. So cabbage butterfly, that is Pyrus brassicae, also known as the large white butterfly. These are the strong flyers and cosmopolitan in distribution. This is the one of the major insect pests of the cabbage to cause serious damage to the crop. These are polyphagous in nature, infesting the different crops like radish, turnip, cauliflower, and other cruciferous crops. Then coming to the entomopathogenic fungus, first time entomopathogenic fungus, that is Beauveria bassiana, reported on silkworm. So these are alternate to the chemical insecticide for controlling the pest of various crops. So this is the natural occurring entomopathogenic fungi capable of attacking and penetration of the host during development. Coming to the mode of action of Beauveria bassiana, the spores of Beauveria bassiana attach to the body surface. After that, germinate and eats the hemolymph, ultimately resulting in the death of the host. Coming to the materials and methods. Here, crop is cabbage and insect pest. I have used 15-star larvae of Pyrus brassicae and entable pathogenic fungus, that is Beauveria bassiana BBJ S1 strain, which is collected from Assam Agriculture University. And here is the rearing of larvae, rearing cage of the Pyrus brassicae, adult food material. We can see here flask-shaped eggs of Pyrus brassicae and different larval instars of Pyrus brassicae. Among these, I have used 15-star larvae of Pyrus brassicae. Here we can see pupa and adults. So we can observe sexual dimorphism. In case of females having 2 black spots on each forewing, coming to the male, only 1 black spot in each forewing. And this is a preparation of Beauveria bassiana solution. After that, culturing of Beauveria bassiana. This is a improved Newbar hemocytometer for counting the total hemocyte count. So coming to the total hemocyte count. So total hemocyte count was determined by the following formula of Jones. That is total number of hemocyte counted in the 4 corner chambers into depth of the chamber into dilution factor by number of chamber counted. Then differential hemocyte count, it was expressed in percentage. Differential hemocyte count equal to N by T. N is the number of particular cell type. T is the total number of cell per unit area. Coming to the results. So here we have observed 4 types of hemocytes, that is prohemocyte, plasmacyte, granulocyte, inocytoid. So prohemocytes, among these cells, prohemocytes are smallest cells and henocytes are large cells. Coming to the nucleus, prohemocytes are having the smallest nucleus and granulocytes are having the large nucleus. And here we can see the hemocytes of the Pyrus brassicae. Prohemocytes are round cell with round nucleus, and plasmacytes spindly shaped with round nucleus, and granulocytes are round shaped with round nucleus, and inocytes are round or ovoid shaped with accentric nucleus. Here we can see treated with Beauveria bassiana against the Pyrus brassicae. This is the control plate. Coming to the total hemocyte count, compared to the untreated insects, here we can see total hemocyte count is reduced with the increasing time. Then coming to the prohemocyte count, compared to the untreated one, prohemocyte count also reduced with the increasing time. And coming to the plasmatocyte count also count increased with increasing time. These plasmatocytes and granulocytes, these 2 are main immune cells in insects immune system. And coming to granulocyte count is increased with the increased time. Coming to the inocytoid count, here we can observe compared to the untreated one, inocyte count is decreased with the increased time. So here we can see response of hemocytes to the Beauveria bassiana at 6 hours of treatment, clumping of plasmatocytes and granulocyte, cytoplasmic extension of plasma membrane and disintegration of the plasma membrane and oozing out of the cell contents of the granulocytes. After 12 hours, we can see clumping, disintegration of the plasma membrane, leakage of the cellular contents and cytoplasmic extension of granulocytes. Then at 12 hours of the treatment, 12 hours of treatment, we can see the degranulation and evacuation. After that 36 hours, philopedal elongation and disintegration of cell contents of the granulocytes, evacuation, and 48 hours, we can see the conidia of Beauveria bassiana hyphal growth around the granulocytes. At 72 hours of the treatment, we can see here mycelial growth, hypha of Beauveria bassiana, degradation of the granulocytes, and 96 hours of the treatment, we can see the capsule formation and nodule formation. Finally, conclusion, the results of the present study demonstrated that this Beauveria bassiana is the most versatile group of the fungus which infect the wide range of the insect pests, including the Replopteran species. So this study clearly suggested that Beauveria bassiana could be incorporated in all our overall IPM program, and also understanding the role of hemocytes in insect defense is essential for the development of the bioresearch. Thank you. Thank you.
was a short and fast presentation. So, how can you think that this could be useful to the farming community? I think it is Shravanti.
Yes, ma'am.
Could you throw some light, like how your findings are directly benefiting the farming community?
Ma'am, nowadays insect immune system also important for controlling the different pests, ma'am. Immunology, defense reactions, different like capsule formation, phagocytosis. These are important for different immune reactions in insect biology, ma'am.
So, Vantiji, what monitoring and detection techniques can be used to assess these populations?
Actually, we did research on Assam Agriculture University. There is a cabbage butterfly is a major pest and it infects all the major crops, like all the coal crops. There, northeastern state, it is a major pest. So it is important for controlling these pests. Using Beauveria bassiana, like alternate chemical approach for controlling this pest.
Okay.
Yeah, if there are no queries from the audience, I think we can proceed to the next presentation, please.
नेक्स्ट को बुला लो रोहित.
जी. Lenthagam, are you there?
Yes.
Okay, we can proceed with you, sir.
Okay. Please share the slide so that I can share. Is it visible, sir?
Not yet,
Is
sir.
it visible?
Yeah, it is.
Can I start?
Visible.
Okay. Perfect. Okay. Okay. Good afternoon, all. Today, the title of my presentation is molecular and morphological characterization of non-yeast rice languages for phosphorus deficiency tolerance. So I won't take much time. Coming to the introduction part, soil acidity is one of the major constraints for rice production in Northeast India due to high rainfall and acidic nature of the soil. Most of the available phosphorus has usually fixed, mostly in upland conditions. So to resolve such crisis, breeding and exploration for phosphorus efficient rice variety could be effective solutions. For this, in this study, we screened 80 rice languages from different parts of Northeast using 7 specific markers. Among these 7 markers, we could see like K46-2. These have the highest amplification percentage across the genotypes. So based on this part 1 data, part 1 markers, we classify the genotypes into different clusters. Here, the cluster 1s with Casalan. Casalan is a part 1 positive check. So here we could identify 27 genotypes being positive to part 1 markers. And
Excuse me, sir. So your slides are not moving.
मैडम चेयरमैन, क्या आपको पीपीटी शो होती दिख रही है?
Actually, it is not moving, sir. It is in the first slide only.
Oh, so mine is already moving.
Please, please
sir, don't
move
go
the
on slide mode.
Okay, is it moving now?
No, it is in the first slide.
No, sir.
Introduction slide only. It is there, your name and details, title.
Maybe internet issue then.
Yeah, it takes some time, but you can just try it.
Is it moving now, ma'am?
No,
sir.
It is only your
Sir.
title.
Sir, don't go for PPT mode. Just do it only in that normal way. Like,
Okay.
yes, sir.
Is it visible now?
Yeah.
Yes, sir.
The
Yes,
slide
sir.
is
Yes, sir.
moving.
Yes.
Okay. So can I come to this part then? Okay, shall I start again from the beginning?
No need. You can go ahead, please.
Okay, so this is the cluster analysis based on the PUB1 markers. So here cluster 1, this is the PUB1 positive genotypes group. Here 27 genotypes being found to be positive for PUB1. So population structure analysis, the population will group into 4 subpopulations. So based on this population data, during gene ELISA, the analysis of molecular variance been done. So here we could see like 25% of the variation were attributed to individuals among individual, while 75% of the variation being attributed to among populations. Here we can see like FST, the value of FST among the population were high. This suggests that there is a strong genetic differentiation among the rice landraces in response to low phosphorus, and the gene flow 0.06 suggests that this is low, which means that the land races which the farmer have been cultivating been still maintained, the genetic purity still maintained. And coming to this field validation, of the 80 molecular screening, we have selected 32 genotypes for field validation. So in the field validation, we conducted into 3 different doses of phosphorus, 0, 20, and 40 P2O5 kg P2O5 per hectares. We could see like under deficient condition of phosphorus, there is a delay in flowering and maturity, and like a reduction in productive tillers like field grains and most of the yield contributing characters. And also we could see like phosphorus use efficiency for soon or grain are like decrease as the phosphorus doses increase in the environment. So from this field validation, we could see like phosphorus, the genotype and phosphorus interaction been positive for most of the traits except days to flowering. So although here we could see like there is a delay or delay in flowering, but there is no significant variation. So we based on the phosphorus use efficiency estimation, we could see like some of the rice land races like Bali, Gungul, Kolong, Banglami, Khuzui. Some of these genotypes were found to be highly efficient as compared to the others, and these are mostly PUB1 positive genotypes. And in addition to this, we also work out various stress indices, just like stress tolerance index, and then partial yield reduction based on the normal phosphorus environment and deficient condition. So based on this stress indices, phosphorus uptake and phosphorus use efficiency and grain yield, the genotypes were classified into 3 groups. Here we could see like the first cluster here consists of 8 genotypes, including cassalates. So this group will consider as identify as highly tolerant to low phosphorus or phosphorus deficiency tolerance, and the rest are identified as low tolerant or moderately tolerant. So among these 8 genotypes, including cassalate, these are mostly PUB1 positive, but interestingly, here one genotype then is gopinet, this Salen races. So this is devoid of PUB1 locus or absence of PUB1. So this genotype, although there is no PUB1 locus, it shows partial or part tolerance or yield capacity with most of the PUB1 positive markers. So from this, we could conclude like there is a huge diversity among the land races in response to phosphorus deficiency tolerance, and some of the identified land races in this study could be used as adonal parents in future breeding program, or set genotypes can also be undergo for genetic improvements. And then the impact of phosphorus levels on growth and development highly depends on the genotypes, not on the doses of phosphorus. And then the low P tolerance genotypes such as gopinet, which I have mentioned, which is devoid of the PUB1 locus. This implies that there is a possibility of a novel source of phosphorus tolerance locus in rice, which is yet to be identified. Thank you.
I think because most of our Indian soils are somewhat medium to rich in phosphorus. So location specific or universally the varieties which are suitable throughout the country or a state level would be definitely a good work with respect to rice.
Yes, ma'am.
But how far this particular genetic material will serve for the other locations and all, it should be still focused upon.
Pardon, ma'am?
Because our country soils are mostly medium to high in phosphorus. Like some varieties or land races have been identified. I hope they should be also tested for other locations and soil types also, whether they are suitable or not.
Yes, ma'am. Yes.
And even the genetic material, whatever is identified, that
Yes,
could
ma'am.
be spread to the other areas.
Yes, yes.
In order to have a sustainable pea management.
Yes, ma'am. Yes, ma'am.
Yeah, I think it was a vast work done in terms of the breeding aspects. But from the lab, it has to turn out to the field in order to fetch more benefits to the farming community.
Yes, ma'am.
Yeah, still any doubts from the audience and the participants? So all these were, I think, dendrograms and all were done by you only, sir? All the analysis?
Yes, yes. Everything, all analysis has been done by me. So everything is done by me.
Okay, okay. I think a lot of work has been done in that aspect. Good,
Yes.
good work. I think extensive work.
Thank you, ma'am. Thank you.
Okay.
Can I stop sharing the slide, ma'am?
Yeah, yeah. Your slide
Okay,
is the conclusion. I think it has come to the end.
Thank you. Thank you.
Okay. I think the next person, last person could also start ahead with sharing screen.
Dr. Gokul, are you there?
Hello?
Dr. Gokul, are you there, sir?
Hello? Hello?
Yes, sir. You can proceed, sir. We can proceed with you.
Okay. Thank whether my slides are moving? Hello?
Yeah, they are visible, please. Sides are visible and you are
Not
audible.
moving, sir. Not moving. They are visible but not
Visible
moving.
है लेकिन move नहीं हो रही है.
I think it is in
Okay.
the first slide, title slide. Yeah, they are
अब
moving.
ठीक है आप start करिए अब ठीक है.
Okay. Good evening, everyone. I myself, Dr. Gaur Krishna KB, PhD in Agriculture Statistics. I am doing my presentation on scrutinizing future temperature pattern using Sarima, ANN, and Sarima ANN different zones of Kerala. So the contents are introduction, mechanism, methods, and discussion, conclusion, reference. So introduction, the climate is changing in an unpredictable manner throughout the globe. And the fluctuation climate create lot of horrific conditions and have disaster situations. So under these conditions, the prediction of change in climatic conditions is with the maximum precision can help to take necessary precautionary measures to control the situation. So such that temperature is an important weather parameter and the changes in temperature is affecting both flora and fauna present in the entire world. So the mild changes in temperature can drastically affect the life cycle of humans, animals, increase pest and disease incidents, cause drought, crop loss or crop failure, etc. So globally, over the last decade, the temperature has been showing a sudden rise and heat waves alerts are coming throughout the summer season in various parts of India. So the fluctuations in temperatures also affect agriculture and allied sectors, and uneven changes in temperature during the vegetative, flowering, or fruiting period can also affect the yield of crops. So the modeling and forecasting of temperature is very important, and so that by using that, the scientific community can take precautionary measures to decrease the impact of fluctuations in temperature over a specified area. So under this, the main objective of the study is modeling and forecasting of monthly maximum and minimum temperature using SARIMA, artificial neural network, SARIMA ANN in the northern, central and southern zones of Kerala. For the data for 42 years, 1981 to 2022, the monthly temperature data was accessed from different zones to data access viewer, and the analysis was conducted using R software. So this is the method that is first is SARIMA. It was model established by Box Jenkins in 1976. And this is the model of SARIMA model. And the model for fitting SARIMA model, the data must be stationary, and it was done using ADF test, that is augmented Tech-Filler test. The model selection criteria was based on least value for AIC, BIC, and AICC, that is Akeke information criterion, Bayesian information criterion, and modified AIC. So even though if we fit the model, we have to check the stability of the model. It is based on L-Jung-Box test. And this is the SARIMA model, which is somewhat big equation. And this is the simplified form of SARIMA model. And next is artificial neural network. It was developed by Warren and McLaughlin in Waterprints in 1943. And it is considered as branch of artificial intelligence that will include genetic algorithms, reasoning with respect to the cases, and develop expert systems. It is used in diverse fields like speech processing, forecast, state estimation, robotic control, machine vision, etc. And according to Gimre and his co-workers, the basic unit of AN is always the artificial neuron, which is made up of an input layer, associated weights, hidden layer functions, and output layer. So this is the equation for ANN with MLP model, which is I used in the study. So next is a simple, this is an architecture of MLP where this is the input. Our data will be divided into inputs. This is hidden layers. And after that, it will produce the output. So what we need? This is SARIMA ANN. This is a hybrid model. It is combining both SARIMA and ANN in such a way that the linear part of the data will be defined by SARIMA, whereas the non-linear part will be explained by artificial neural network. So the model was evaluated. The evaluation of the model was conducted using MSE, that is mean square error, root mean square error, mean absolute error, and mean absolute percentage error. And the least value for this will consider as the best outperforming model. So next is result and discussion. So the whole data is divided into two sets. That is the 40 years 1981 2020 was placed in the training set, and testing set consists of 2 years 2021 2022. So from this data we can clearly understand that the data, the range of the maximum temperature is highest in central zone. This blue color indicates central zone, and the green color indicates northern zone, that is the second, and the red color indicates northern zone. And it is moving in fluctuating manner over the years. And we can see that the range is almost touching from 25 to, and it is not crossing 40. I'm telling this because after the forecasting, we can see that some changes happening. And also this is the minimum temperature. In minimum temperature, the highest minimum temperature is shown in the red color, that is northern zone, and the least is shown in central zone. And the green color is showing somewhat middle nature. So next is the descriptive statistics. So maximum temperature, minimum temperature, as I told, the figure is clearly explaining the same thing. That is the maximum values are obtained in the central zone, and the minimum values in northern for maximum temperature. Whereas the minimum temperature, the central zone is showing least, whereas northern is showing the highest. Southern is almost showing the constant manner, that is median nature. And these are the SARIMA model best selected for the each zone and for the maximum and minimum temperature. And this is the parameters. This is the criterion and these values. And this checks the stability. The stability, the put forward that for the maximum temperature, the SARIMA model fitted are good. But in minimum temperature, the central and southern zones, since this is significant, that means our model is not stable. So we cannot say that it is a good model. But since we are using other models, we can just compare whether it is showing, even though with the presence of instability, whether they are showing good in forecasting. So this is AN architecture. We can see that for the whole actual data, the architecture consists of input is some 22 to 21, 23, 22 to 23, because the values will be exactly this original data. Whereas for the residuals means the residuals obtained from the SARIMA, that is actual minus fitted. For that, we can see that it is 11, 10, 12, 11, 12, like that is the range for input nodes. Whereas the hidden layers and output will be same for all. So after that, we will do the validation. The validation suggested that for the northern zone, it is artificial neural network, and also the southern zone, it is artificial neural network for the maximum temperature. Whereas the central zone, where the temperature was high, it was SARIMA model which have high outperformed other models. Whereas in minimum temperature, northern zone showed the maximum values, and there it is SARIMA AM showed the higher efficiency in prediction of the model. Whereas in central, it is ANN model, and southern also it is ANN model. And next is forecasting. So prediction of monthly maximum and minimum temperature for next 5 years, that is 2023 to 2027. You can see that here the peaks are touching 40. So that means before 1983 to 2020 we compare, it was not that much touching to 40, but the future is considered as the prediction of the model, it will be considered as it will be showing almost 40 in the central zone. And as it is 2020 2024, now we are in the summer season. This 40 degrees exactly now in the central part of my state, that is Kerala, it's showing exactly this 40 above degrees Celsius. And also,
sir, sorry to interrupt you in between. Sir, sorry to interrupt you in between. Please conclude within 30 seconds, sir.
Sir, this is my last slide. Next is conclusion, sir.
Okay. Okay, sir.
Okay. And this is the lowest, that northern zone have somewhat less temperature like that. And for minimum temperature, northern is showing increase. There also in the original data or the data, we can see that the values are there in between 15 and all, but the prediction is telling that 15 and all will not be there. It will be increased to 18. There will be sufficient increase in rise of level of temperature over the regions. Like that, the prediction is telling that all outperformed model is telling that there will be increase in temperature, both maximum and minimum temperature, or all the 3 zones in Kerala. So my conclusion is the climate change affect the growth and development of flora and fauna. Anticipation of Twitter pattern weather parameters help next take necessary precaution steps to overcome the impact. So the main focus was to current research work was to determine the future pattern of maximum minimum temperature in different zones of Kerala for the 42 years, and the different models were SARIMA AN and hybrid SARIMA AN models. The results suggested that for maximum temperature, the most efficient model in the anticipation is AN in northern and southern zones, whereas SARIMA in the central zone. The outcome also advocate that AN outperformed other models in forecasting minimum temperature in central and southern zone of Kerala, whereas SARIMA AN in the northern zone of Kerala. The outperformed models were used to forecast future temperature for next 5 years. The anticipated values suggested that there is a rise in minimum and maximum temperature for next 5 years. So the scientific community, as we all belong to the different scientific community, must produce new innovative techniques to adapt the rise in temperatures so that it may not affect the growth and development of flora and fauna, especially in plants. We know that we human can adjust the temperature. That means if the temperature high at 11 to 3 means we don't want to go to the field, we can decide like that. But the plants are there in the field and it cannot move anywhere. Only it can grow. It have to face this situation. So we have to take novel techniques. We have to develop new novel techniques so that the rising temperature can be the rising temperature cannot affect the yield of our crops and also quality of the crops, quality of the yield, quality of the productions. So reference and thank you.
I hope the study was good, but if any correlation was done with the plantation crops and the effect of temperatures on that, it would be more reliable.
Yes, ma'am, but my study was modeling the weather parameters as a whole. My PhD was full of weather parameters and 3 zones like that.
Yeah, but if any crop, because it is agriculture based, that required only some data on crop, that would have been really fantastic for the farming community, so that you can correlate the effect on the performance of the crops.
Yes, ma'am.
Otherwise, as a agriculture sector, we generally correlate everything with the crops. With a mere increase in temperature or a decrease, we cannot correlate it with anything. The work is good, but a little extension is required.
Okay.
So that you can collaborate with some, horticulturist or someone else, so that you can work.
Why there is a difference in efficiency of these models in different regions? So
yes, ma'am.
Why there is a difference in efficiency?
Ma'am, fluctuation is more. Even it is the same state that different regions have their own climatic conditions. The central part of Kerala is very high temperature, whereas the southern is somewhat medium temperature. By north, it is like almost constant. They will behave like that. North, the temperature is somewhat bearable. But central part means it is like a heavy temperature zone area. Within the state also there is very drastic difference is there. Because of that, the same model cannot show the fluctuations are the main reasons.
Sir, what advantages does this Sarima approach offer over the traditional models for temperature forecasting, sir?
Sarima model is mainly for the seasonality. The weather always have seasonality, ma'am. That means in winter the temperature will go down, whereas also in summer the temperature will go rise. Sarima always catches that. Because of that, Sarima is highly implemented in weather parameters. Whereas the artificial neural network is like we can use in any complex data, we can use artificial neural network. It can always collect. It always deals with complex data.
Okay, thank you.
Okay.
अब रोहित जी देखो जरा कितने बचे हैं अब।
सर 2 ओरल के बचे हैं एंड 5 पोस्टर प्रेजेंटेशन के हैं।
ठीक है, ओरल के कराओ यार, 5-5 मिनट में खत्म
जी जी जी। और गंगाधर आर यू देयर सर? गंगाधर सर और मैम सर, हेलो। Anupama ma'am, are you there?
Hello, sir.
Okay, Gangadhar sir, yeah, we can proceed with you, sir.
Okay, sir. Can I share, sir?
Okay. Yeah, you can.
गंगाधर तुम्हें तो यार एक्टिव रहना चाहिए।
हाँ सर, आई एम शेयरिंग सर।
करो करो करो, जल्दी करो।
Good evening to all. Myself Gangadhar Dogoli. I am a PhD scholar from Department of Genetics and Plant Breeding, US Dharwad. And I am going to present on the assessment of genetic diversity and its relationship for heterosis for forest rights in maize. And maize is a queen of cereals, and it's an introduction part. So it's also grown for a grain purpose and as well as fodder purpose. It's a seed plants, and high genetic diversity is there as per the morphological characters, and it's also high seed multiplication ratio also. And US is the toppest growing country for maize, and among in the Karnataka, among in the India, Karnataka is also one of the major maize growing state. So main objective for my presentation is assessment of genetic diversity in inbreed lines for forage traits in maize. And for assessment of the genetic diversity for forage traits, it's done in work was done in VC4 India. So we took 54 inbreed lines for diversity purpose, and it is shown in the row length of 3 meter length and 2 replications. And these are all the lines for we took for diversity analysis purpose. And these are 54 lines were developed in 50 among 54, 50 lines were developed in the VC4 India, Karnataka. And these are all the phenotypic observations recorded for diversity analysis. 50% of tasseling and 50% silking, plant height and number of leaves per plant, leaf length, leaf width, and leaf stem ratio is most important for forest traits. And one more important trait for phenotypic observations for diversity is green fodder yield and also dry matter yield per plant. So experimental results for this diversity analysis. So for diversity analysis we analyzed it using the Mahalanobis D-square method, and among that we took observations for phenotypically observations. These are all the ratios are mean for the phenotypic observations, and among 54 lines we mentioned for the 50% tasseling. Lowest is important for early maturity type. That's why in that among the 54 lines, 1-20-1 is showing 42 days for 50% hazeling. And for our main trait is green fodder yield per plant. So 5-2-1-12 is showing the 116 2 gram per plant in lowest, and highest is 5-6-1. It's showing the 514.87 grams per plant. So it's the green fodder yield is per plant is so most important for forage traits. That's why these are all the this is the line showing the highest green fodder yield.
Please move the slides. It's not moving.
And percentage of contribution for the percentage of the trait, which is contribution for the diversity analysis. So we given the rank among the contribution of the contribution of the diversity. So green fodder is showing the 73% for contribution for the diversity, total diversity, and least for the green matter content. So our trait is green for forage yield is green fodder yield. So it's showing the positive for the diversity analysis. And this is the cluster. This is the clusters based on the cluster distance, based on the diversity, based on the analysis of
Slide
square.
not moving, please. I hope everyone has got the same thing. It is in the first slide only, please.
It's not moving, ma'am.
वेलकम आ रहा है इनका, बस वेलकम ही कर रहे हैं ये। गंगाधर थोड़ा आगे भी बढ़ो यार।
सर it's moving, sir.
नहीं मूव नहीं कर रही है आपकी पीपीटी
अभी सर।
हाँ अब ठीक है, अब ठीक है। हाँ आप बोलिए।
ओके सर। This is table number 4. This is the intro and inter cluster distance for different based on the Mayer-Gomez D-square analysis. We seven number of clusters were arrived. So based on the distance analysis, so there will be the more distance between the cluster number 4 and cluster number 6. So it will indicate that. So if the more is the distance between the between the clusters, that will indicate that there will be a more vigor of more hybrid vigor. So if you cross between these two clusters, we'll get a superior transgressive. So this is the grouping of the 54 inbuilt lines for 54 inbuilt lines. Among this one, we got cluster number 2. We got 18 number of genotypes. Cluster number 2 comes 18 number of genotypes comes under cluster number 2, and maximum number of genotypes comes under the cluster number 2. And cluster mean analysis for different qualitative traits. So cluster number 4 is having the more number of qualitative traits. So it will indicate that so cluster could be selecting for the parent desirable hybridization program, because it's having showing the more number of qualitative traits for further purpose. And this is the dendrogram. Which is given, which is showing the 7 number of clusters. Thank you, sir.
What is the recommendation that you want to go ahead with the farming community, please?
Huh?
What is that you want to recommend to the farming community?
On what?
Out of your results and all. In a
Yes, ma'am.
point.
Actually, ma'am, as I shown here, cluster number 4 and here. This is a cluster number 4 and cluster number 6, ma'am. If you crossing between the cluster number 4 and 6, we'll actually we got a good number of hybrid lines, hybrids, ma'am, around 200 hybrids. Among that, we got 5 superior hybrids, ma'am. For powder purpose, farmer can use that hybrids for their fodder purpose to feed the cattle.
Yeah, because these were all completely breeding trials. I just asked in one word. That's why, in a simple way, what could be the outcome to the farmers?
Ma'am?
Yeah. Based on this, you have identified those two lines in terms of order lines. Okay.
Yes, ma'am.
Okay. Okay. Still any questions from audience? Otherwise, I think one more person is there for presenting.
Anupam ma'am, are you there?
Sir, tell me, sir.
Yes, sir. Yes, sir.
Okay, we can I think proceed with you.
Please stop sharing.
Okay, thank you, ma'am. Thank you so much.
Sir, my slides is visible?
Yes, ma'am.
Oh, hello everyone, myself Anupama. I'm the PhD scholar from CU Infan, and the title of this my presentation is Effect of Different Nutrient Management Practices on Black Aromatized Nutrient Content, Nutrient After and Soil Chemical Properties under Black Aromatized Broadband Cropping Sequence. So, right is the lack of 4 millions of people and stable for more than half of the population. The black center rice in Manipur, we locally know as Sahau, its great qualities are the color and the aroma, along with the sweetness, is not common in other part of India. And black aromatic rice is considered of the research source of anthocyanin. Founded, but apart from this, it also have high content of vitamins, mineral, fiber, protein, and the nutrients. And then over and use of chemical fertilizer result in human health soil hazard, which affect our ecosystem. So many researchers has found that neither inorganic or the organic sources can result sustainable productivity. So the sustainable productions of this black raw materials of Manipur can achieve by adopting the integrated nutrient management system. So objective of the study is to find out the effect of nutrient management in current black raw materials, nutrient content, nutrient after some chemical properties. So design of my experiment is for Kharif. Rice have RBD design with a treatment replication. The variety is Poreton with a seed of 40 kg per hectare. And for the Rabi season, design is split plot design. Main plot is the residual effect with 8 and sub plot is 3. And this broadbean variety is local variety with a seed of 25 kg per hectare. And this is the treatment detail for my Kharif rice experiment. And for Rabi season, the main plot is the residual effect of the Kharif season treatment. I have 3 subplot. One is control. There is no fertilizer application. Say F1 is the 50% RDF and F2 is the 100% RDF. This is the field view, my research, and some photos. And this so the effect of nutrient management on NPK content to rise. Here the nitrogen content is high in the N2, but which is at par with the N5. And also the lowest content of nitrogen is in the organic treatment, and but which is at par with the N7. And in the phosphorus content in the seed also, the highest content is in the N2, it also par with the N5. And the lowest content is in the N8, but it is at par with the N7 and also with the N4. And coming to the potassium, significantly higher presence of the nutrients present in the N2 and the lowest is in the N8 treatment. And coming to NPK content on the stopper of the amateur rice, nitrogen content is highest in the N2, but it is par with the N5. And lowest is in the organic treatments, is N8. And coming to phosphorus content stopper, the significantly higher content phosphorus in the N2, but lowest in the N8, but it is at the par with the N7. And in coming to potassium, the highest in the N2, but it is part with the N5. And then the lowest content of potassium is in the N8. And coming to the total uptake by the black materials, here the nitrogen grade uptake and nitrogen store uptake is higher in the N2 and is par with the N5. But the lowest is in the N8, it is par with the N7 and the N4. Similar also follow in the total nitrogen uptake by the grain and the store of the blood meteorites. And coming to total phosphorus uptake by the blood meteorites, it also phosphorus content by straw by the grain also high in the N2, but the lowest is in the N8, but it has found par with the N7, and also similar trend also found in the phosphorus total uptake. And then this effect of total potassium, this significantly highest potassium content is in the N2 treatment, and lowest in this organic treatment is the N8. And coming to the about the available NPK organic content after the harvest of black rice, the highest available nitrogen is found in the N2 in the both year. N2 is the highest nitrogen content, but it's par with the N5, and the lowest is in the N1. Is the RDF 100% RDF, but in the pool data, highest content is in the N5 and lowest is in the N1. There's no par. In the coming to the phosphorus, this highest content of the phosphorus is in the N5, this par with the N2, and the lowest in the N1. But this par between the two positions, but in the pool there's no par. And coming to potassium, this highest potassium content also in the N5, but it is par with the N2 in the both year, and lowest in the N1. And coming to organic carbon content, highest organic carbon content is in the organic treatments, and lowest content is the N1, is 100% recommended of the fertilizers. And its conclusion that after 2 years of experiment, this N2 is 100% RGF, versus Banya azospamine powder recorded maximum number of nutrient contents due to uptakes by blood meteorites, and highest soil available NPK was observed in the N5, and NI this organic treatment have recorded the highest organic carbon content. Thank you, sir. Hello?
I have one doubt, like, for this, because you have given rice and broad bean and that to nutrient management, like in aromatic rice, what is the role of like organics in improvising upon these quality parameters?
Then organic rice, they have increased nutrient content in the plant, and coming to the soil, they improve the soil physical and chemical properties, increase microorganism also, that will increase the yield of the crops.
So your climate, I think your climate is also very congenial for aromatic rice.
Yes.
Yes, I am audible. Your climate at Impal is congenial for cultivation of aromatic rice. That is suitable.
Yes, ma'am, suitable. Yes, ma'am, suitable.
Okay, then. So broad bean, like you have told, along with this, what are the other crops which are grown with rice-based cropping system?
Well, mostly here we grow pulses like lentil, also pea, and like mustard. We grow with mostly with the rice, ma'am.
Oh.
In our research farming.
So out of all this, do you think broad bean, rice followed by broad bean is better over them?
Ma'am, it's not better. I think that here I can say it's better. But I think ma'am here though more like suitable is pea. Pea is more suitable. But I'm trying in broad bean. It's my first time.
I think we have come to an end of the oral presentations with this, sir.
Ma'am, we have like last participant is left.
Okay. Shall
Just
I go
last.
ahead after this, my two presentations?
Ma'am, can I present my slide? Sorry, am I audible?
Yes, madam.
Yes, ma'am. Yes, ma'am.
You are audible.
Okay. Good evening, everyone. I was respected chairman, co-chairman of this international seminar. I'm Dr. Bharati, scientist from ICR Research Complex, Mizoram Center, Policy. Title of my presentation is Yield and Economics of Ginger
The voice is very low. We are not audible.
The title of my presentation is Yield and Economics.
Little voice could be raised. That's all.
The title of my presentation is Yield and Economics of Ginger Intercropping System, and this is the research under AICRP SPICE. As we all know, ma'am, am I audible? Hello?
Yes, ma'am. Yes, ma'am. Audible.
Ginger.
Yeah, audible, madam. Little raise in voice. How you have done it is enough.
Okay. As we know, ginger is a major spice crop of Mizoram with huge economic importance, and the climate condition of Mizoram is highly suitable for quality production of ginger. And as we see, the production productivity is 60 metric tons and 7.03 metric tons per hectare. And it is ginger is a shade-loving crop and requires ample moisture for growth and development. And it is suitable crop for growing with other crops for better yield. And the traditional practice of farmers in Mizoram, it has been traditional practice among the Mizoram farmers with beans, taro, maize, banana, and papaya. And although ginger-based intercropping is followed for many years, much success has not been achieved by farmers in crop production and productivity because there is lack of scientific knowledge in crop cultivation. There is improper selection of intercrops. There is use of poor knowledge in quality seed with regards to quality seeds, lack of crop and soil health management, etc. And also farmers are unaware of lucrative profits that can be earned through complementary use of crops to generate income. And practice of intercropping throughout the year offer great scope. And ginger-based intercropping system improve crop yield and better returns. And so it is crucial to identify suitable ginger-based intercropping system for improving crop yield and ultimately income of the farmers. So the objective of my experiment is to identify suitable ginger-based intercropping system to get better rhizome yield and system production, and to study the economics of ginger-based intercropping system, and to identify best combination for recommendation to farmers in terms of better results. And these are the treatment materials. 7 treatment comprised of sole ginger, and treatment 2 is a combination of ginger, fenugreek. 2:2 ratio means 2 lines ginger intercrop with 2 lines fenugreek, and accordingly. And treatment 3 comprises of ginger, coriander, and leafy vegetables in 2:2 ratio. And treatment number 4 is ginger and maize in 2:2 ratio. As we see, T5 is ginger and French bean in 2:2 ratio. And treatment number 6 is ginger and arhar in 3:1 ratio. And treatment number 7 is ginger and taro in 2:2 intercropping system. And the experiment was conducted in research from the ICR Mizoram Center. And the design of the experiment is randomized block design with 2 replication. And these are the observations recorded for the experiment. I will not go in very detail. Treatments recorded are plant height, number of tillers, fresh weight of plum, number of rhizomes, ginger yield, and intercrop yield, crop equivalent yield, gross income, net income, and benefit cost ratio. And this is my results in one slide. From the table, we can see that maximum ginger plant height, that is 68.23, and fresh weight of plum, that is 192 gram, was recorded in ginger and French bean intercrop in 2:2 ratio. And whereas ginger and French bean intercrop also significantly increased the ginger rhizome yield, which was 9.7 higher compared to the sole ginger. As we see from the table, these are the table presenting the plant height, number of tillers, fresh weight of clump, number of rhizome, ginger yield, first intercrop yield, second intercrop yield, in which treatment number 5 gives significantly higher results. These are our experimental plot. First one is with sole ginger, and this is ginger and fenugreek. This is ginger, coriander, and leafy vegetables, pak choy. This is ginger maize intercrop. This is ginger French bean intercrop. This is ginger arhar, ginger taro, and so I want to conclude from this experiment that ginger and French bean and ginger and maize in ginger French bean was found to be better with respect to rhizome yield, and whereas ginger maize, ginger coriander, and leafy vegetable intercrop gives better BC benefit cost ratio return. So yeah, in one line, ma'am, we can say that for the farmers in Mizoram, this ginger French bean intercrop and ginger maize, coriander, and leafy vegetable intercrop fetch a higher return compared to other intercrops. Thank you.
Madam, because ginger maybe underground and tuberous crop, like out of all the crops which you have grown, which would do you like to recommend at the farming community based on the, what to say, BC ratio and performance?
If I have to recommend one intercrop, that would be ginger and French bean, which would, because it gives higher yield, best yield, ma'am.
But finally, based on the economics and benefit cost ratio only, we will recommend. That's why I was doubtful. That is giving 3.80, highest out of all the intercropping sequence.
Yeah.
Based on the price and all.
Yes. So
that should be recommended, because in your conclusion, you have to write only one sequence based on the benefit cost ratio.
Yes. So yeah, with respect to yield, this ginger and French bean give, but with respect to BC ratio, ginger and maize gives best result.
That's what only. That's why
yes,
we finally give our recommendation based on the economics.
Yes, that's right. Thank you.
Okay.
क्या हुआ रोहित जी, इंटरप्शन कैसे हो गया ये?
हेलो सर।
हाँ, क्या हुआ एकदम से सुनसान?
पता नहीं सर, आई थिंक कंप्लीट हो गया। अभी मैम बोल रहे थे उनको प्रेजेंटेशन देने के लिए।
अच्छा, तो मैडम का बचा है शायद, भानुरेखा मैडम का। मैडम आप करेंगे क्या प्रेजेंट? मैडम सुन पा रहे हैं आप?
Is there any presentation left out, sir?
नो 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 मैम, नो मैम।
बस मैम आपका बचा है, आप चेयरमैन खुद प्रेजेंट करेंगे।
How many poster presentations are there, sir?
Like there are 20, ma'am. Like.
Okay. I think we will wind up with this oral presentation, sir.
यदि मेरी रिक्वेस्ट आप मानते तो केवल 5 पोस्टर प्रेजेंटेशन करा लेते, फिर वाइंड अप कर देते। आप हार्डली 15 मिनट लगेगा।
नहीं नहीं सर, मैं क्या बोल रही, मेरा भी will you give me chance to present? I was asking that one.
अच्छा,
हाँ,
तो
or else
आप
if you
करना
want,
चाहेंगे?
I am in front of system only sir, so that one session will be
ठीक है,
complete.
प्लीज कर दीजिए मैम, कर दीजिए।
Okay, okay sir, I am sharing, trying to share.
ओके।
Sir, is the voice audible sir? Am I audible sir?
Yes, ma'am.
Yeah, I am audible. My slide is visible madam?
हाँ, yes madam, visible. Please continue.
I am going to present on the performance of machine planted chickpea under varied seed rate and nutrient management, sir. This is my PhD student work, and I was the chairman. On behalf of her, I am presenting, sir. So, coming to the background and all. So, as we know, pulses are the versatile crops, and they have a lot to do with the sustainability of the soil and to reduce the inorganic fertilizer requirement. Since they are very important in food and nutritional security, aptly called as poor man's meat, as they contribute a lot to the protein requirement. And among the pulses, chickpea dominates with a 49% share of total pulses, and it accounts to 50% of the total pulse production. And a major part of the vegan protein, superior to other pulses. And on account of the mineral content, nutrients, and other growth parameters and all, it has also a strong role to play in reducing the chronic diseases like cardiovascular diseases and so on. And hence it is aptly called as functional food among all the pulses. And if we observe, pulses are such a crops that they are grown on submarginal and marginal crops, and the harvest index as well as the post-harvest losses are very high. Apart from that, the requirement of the labor is also very high. With the reduction in the labor and hike in the prices, there is a need to switch over to the mechanization in all the pulses, which require a lot of practices from sowing to harvest. And even Indian Institute of Pulse Research, based on the Vision 2050, it has given a strong step that mechanization is the most important thing in order to increase the production, productivity, as well as to be self-reliant in the pulse production. So when we switch over to the mechanization, that is sowing of the mechanization and all, the first and foremost thing is that we need to standardize what is the population or seed rate required, and accordingly the fertilizer management is also very important. And these two hold a prime position in the agronomic practices, and we have taken this point as a base of our study, and we have framed a study on the chickpea with partial mechanization. Sowing was done with it, and some of the operations like weeding with the power weeder and even harvesting with the reaping we have done, keeping these points in view as some of our objectives. And coming to the nutrient management, increased inorganics have already proven that they have detrimental effect on the soil as well as on the the environment. So we have taken microbial consortia as one of the component in the nutrient management as a subplot. So we had two treatments, that is main plot was seed rate and the subplot was the nutrient management practices in chickpea. So I'm focusing on the technical details. I hope the slides are moving ahead.
Yes, ma'am.
Yes, madam. Yes, madam.
The technical details are in the Rabi season, we have taken 2 consecutive seasons, that is 2020-21 and 21-22 being the PhD study. And the location was at Agriculture Research Institute main farm with 28 treatment combinations, and the design was split plot with 3 replications. So in the main plot, you could have a eye on this. That is, we have taken 4 seed rates according to different plant populations. And this seed rate we have arrived after calibrating with the CIAE, that is Central Institute of Agricultural Engineering, Bhopal planter. And we have taken the 4 seed rates with this planter. That is 52 kgs, which accounted for a plant population of 2.22 lakh, 70 kgs with 2.96, 77 kgs with 3.33 lakh, which is recommended by hand sowing, that is manual sowing, but the seed rate was less here because it was with the machine. And 105 kgs, that it had a little more population. That is, generally we go for 100 kgs with the manual sowing, whereas with the 105 kg, it accounted to 4.44 lakh. And in the subplots, we had different nutrient management practices like absolute control, 75% of the RDF, 100% and 125%. Whereas we also had a combination with the microbial consortia, that is Estobacter, phosphorus solubilizing bacteria, and KRB, which was from our PJTSA only. That is, we have a biofertilizer unit. From there, we have taken at a rate of 5 kgs per soil application with 200 kgs of FIM or compost. So the next 3 treatments, that is from S5 to S7, consisted of 75% RDF plus this 5 kgs of consortia, 100% with consortia, and 125% with consortia. So when we have a look at the technical details, like the season was Rabi, and the treatments are 4 into 7, that is 28, and the design was split plot, main plots, the seed rate, and subplots, nutrient management. And the spacing for this respective populations, whatever we had taken, was 30 by 7.5 and 30 by 10, 45 into 7.5 and 45 into 10. And the variety we have adopted was the most famous variety, the farming community, JG11, and the soil type was vertisol. As we have already discussed, that was CIAE planter, which had a shoe type of opener. And when we have a glance at the yield attributes like pods per plant and the seed yield of chickpea, so we could observe that among the different seed rates, the seed rate of 105 kgs, that is M4, 105 kgs per hectare at 4.44 lakh population. And among the nutrient management practices, the S7, that is
कुछ नेटवर्क में प्रॉब्लम आ रहा है आई थिंक।
शायद सर।
हाँ हाँ, मैडम के मॉडरेटर ठीक भी कर रहे हैं उसको। हेलो मैडम, कुछ नेटवर्क में प्रॉब्लम आ रहा है आपकी साइड से।
Sir, am I audible? I have connected through net data. Am I
ठीक है
audible
मैडम,
now?
ठीक है, कंटिन्यू करिए आप, प्लीज कंटिन्यू करिए।
Yeah, we had a power cut, sir. I have joined through the mobile network.
नो इशू मैडम, नो इशू, नो इशू।
Yeah. So as we have explained, if I am audible, I think it is okay, sir.
Yes, 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 yes. Please continue.
The seed rate of 105 kgs per hectare and the nutrient management 125% with 25% of 5 kgs microbial consortia has resulted in higher yield as well as pods. And when we see the protein yield, gross returns, net returns, and BC ratio, the same treatment, that is 105 kgs per hectare and 125 kgs RDF plus 5 kgs consortia has performed better. And the same thing I have concluded the same. But in the second year, interestingly, the interaction was significant. 125 kgs RDF plus 105 kgs seed rate. Whereas in the first year, individually they have performed well. That is main plot 105 kgs and subplot. Whereas in the second year, the interaction was very good. And in both the years, the benefit cost ratio was high with 105 kgs seed rate and 100% to 125% RDF, which were on par with each other. So we have given a recommendation of seed rate of 105 kgs with what we say 100% RDF, and microbial consortia was better with a saving of 25% of fertilizer from our study. This was the outcome, sir. We have even references. Like I have given 8 references and different pictures of our what we say the field experiment. So this comes to an end, sir.
जी मैम, मेरा एक छोटा सा क्वेश्चन है मैडम.
Yes, sir. Please.
कि पल्सेस के फील्ड में हम आत्मनिर्भर कब बन पाएंगे?
The only thing is that, sir, it should be taken on a larger areas by the farmers for mechanization. And they give very less importance because in Rabi and all they grow, and they do mostly as intercropping and so on. But if you want to go for mechanization, lot of area should be there under this with large plots and so on. Otherwise, they give more importance to cotton, soybean and all in Kharif season, whereas coming to the Rabi season with little amount of irrigation, whatever is there, they go hardly for small patches where mechanization is not possible.
बहुत बढ़िया प्रेजेंटेशन, तो अपने आप में सेल्फ एक्सप्लेनेटरी था, उसमें क्वेश्चंस बनते ही नहीं है. मैडम हर्षदीप, आप कुछ पूछना चाहेंगे? मैडम हर्षदीप.
No, sir, the presentation was good.
ठीक है. क्या हम कोई पूछना चाहे? यदि क्वेश्चन कुछ किसी के जहन में, किसी के मन में हो तो पूछता है. नहीं तो क्या मैडम हम पोस्टर की तरफ मूव करें?
Yes, sir. Actually, I have one more presentation, sir. That is my another student. But I think we will start with the poster, sir.
Sir, one question is there.
Whether-
Ma'am, one question is there in chat box. What is the
Yes,
seed
yes,
rate
what is
with
that?
sowing time?
Yeah, seed rate, we had 4 seed rates, madam. The sowing time was, we have taken in November 15th to 20th. That is Rabi, because it is a Rabi crop. And we had four seed rates in the mechanical planting. Out of the 52 kg, 70 and 77, 105. 105 kg seed rate per hectare was performing well. And seed rate was, sowing date was uniform. That is November 2nd week, November 15th to 20th.
Okay.
Sir, whether we will be completing the posters today or tomorrow also there is session, sir?
एक्चुअली मैम 5:15 आज हो गया है, तो हम सोच रहे हैं 5 प्रेजेंटेशन आज हो जाए, बाकी कल कर लेंगे.
Okay, sir. Then the second presentation of my oral tomorrow I'll give. We'll start with the
ठीक है,
poster.
तुम्हारो कर लेंगे, कल कर लेंगे. कल भी मैम समय निकालिएगा, थोड़ा टाइम निकालिएगा
You
थोड़ा.
tell me the time, sir. Not an issue. I'll come off to the office. It is nearer to my listing one.
ठीक है, थैंक यू मैडम. रोहित जी, पोस्टर के लिए आप मूव करिए.
Yes, sir. Hello, I'm Mona Lisa Devi, ma'am. Are you there, ma'am?
Yes, sir. Yes, sir.
Okay, we can proceed with you, ma'am, for the poster.
Okay. Sir, is my slide visible? Hello?
अभी तो विजिबल नहीं है, अभी नहीं है विजिबल.
It is just sharing, madam.
Yeah, I have shared, ma'am.
It's not visual, ma'am.
Sir, is it visible right now?
No, ma'am.
Yes, visible.
Sir, is it visible right now?
Yes, ma'am.
Okay. There's some technical issue, that's why. Okay, good afternoon, everyone. Today, I'm going to present a poster presentation on the effect of biopesticides on eggs length and fecundity of adult female of Chrysoperla jastrovillicelmi in the subsequent generation. Myself, Moirangthe Monarisa Devi, and I'm pursuing my PhD in College of Agriculture, Imphal. So in introduction, biological control, as you all know, is an environmental friendly methods of control in which green lacewing Chrysoperla jastrovillicelmi has a significant role to play. So Chrysoperla jastrovillicelmi has been proven effective in field crops, orchards, and greenhouses. And moreover, entomopathogenic microbial fungi, for example, Bibera vacina, Bacillus thuringiensis, Metarism anisopli, has played an important role. So there is a need of when we choose them, we have to be we have to keep in mind that they have a low toxicity in the Chrysoperla jastrovillicelmi. So let's move on to the methods and methodology. So for rearing the grafts of the Chrysoperla jastrovillicelmi, Corsera cephalonica eggs, which were procured from the biological control lab in the OUT Bhubaneswar, So from there, the mortality effect of the biopesticide of the third instar larval stages of the current generation were investigated by adopting the two methods, that is the residue deposit method and the dyed contamination method. In residue deposit method, the glass petri dishes were sprayed with different treatments separately at the maximum recommended doses. And in dyed contamination method, UV treated Corsera eggs were sprayed with different treatments separately at the maximum fill recommended doses. After that, the larval mortality in both the methods were observed at 3rd, 5th, and 7th days after a treatment, and the mortality percentage were worked out. So the survived population of the Chrysoperla jastrovillicelmi in the mortality study were selected for the study of the biology in the subsequent generation, where the survived larvae were reared until pupal stages, and then pupae were individualized into the separate biles followed by the adult emergence. After that, the emerged adults were paired in male and female and they are kept in a separate plastic jars. So after that, the adults which are reared and produce the next generation, and from that, the egg length and the fecundity of the adult female of the Chrysoperla jastrovillicelmi in the subsequent generation was observed. So let's move on to the result. In the treatments, Bacillus thuringiensis, Lecanicillium nicani, Bibera vacina, Metarism anisopli, azadirachtin, fipronil, and the untreated control, these are the treatments that have used in the experiment. And so in the residue deposit method, the egg length, the maximum egg length was observed in Bacillus thuringiensis, that is 1.71 millimeter, that is which is at par with the untreated control, that is 1.71. Here we can see in the slide. So in the lowest can be seen in the fipronil treated larva, and in dyed contamination method, the maximum length was observed in Lecanicillium lecani, that is 1.64, same as that in untreated control, which is at par with this. So, and the minimum was shown in fipronil. So in the next table, we can see the fecundity of the adult female. So the highest fecundity was observed in Bacillus thuringiensis treated larva, which is at par with the untreated control, and the minimum was observed in fipronil treated. Likewise, in that contamination method, the highest fecundity was observed in Lecanicilum lecani, that is 311.67, that is in terms of number, and then lowest was seen in fipronil. So from these tables, we can conclude that the microbial pesticide has little or no effect on the egg length and the fecundity of the adult female of Chrysoperla jastrobicillemi in the subsequent generation. So as a whole, the microbial pesticide has no significant effect on the biocontrol agent Chrysoperla jastrobicillemi, which can easily depicted that they can go hand in hand as a set in IPM packages. Thank you. Hello, ma'am.
Madam Bhanu Rekha.
So you have given the egg parasitoids. So apart from this, you told that it has not so much control on this, isn't it? On the egg length and
yes, pardon, ma'am.
You told that that is not so effective, isn't it?
Ma'am means like the biopesticides that I have been used in the experiment has no effect on the Chrysoperla jastrobicillemi.
So whether it is useful result or you say it cannot be taken to the farming community.
No, ma'am. Actually, in IPM packages nowadays, like most of the people or the farmers, we are been dealing with the integrated pest management. So people are using microbial pesticide as well as the biocontrol agent like the Chrysoperla jastrobicillemi. Actually, this Chrysoperla jastrobicillemi is a biocontrol agent that their grubs feed on the aphids of the crops. So when using together, farmer got confused that like when we use the microbial pesticide, will it be effect to the biocontrol agent that is Chrysoperla jastrobicillemi? So there is a question. So in this experiment, we conclude that there is no effect for this microbial pesticide to that other biocontrol. So we can use both of them as an IPM package for controlling the insects.
I think this is a good result with which farmers can get convinced in using microbial pesticides over inorganics. But
yes,
what are the drawbacks? Sometimes what are the drawbacks with these microbial pesticides? Are they working at a different level of the infestation? When the infestation is very high, can we completely rely on these biopesticides?
Actually, ma'am, there is a hindrance in getting because the price is also very high while comparing to the other. This one procurement is procurement for this microbial biopesticide is a little bit hindrance to the farmer. And then the thing is like it's a very slow process when we compare with the chemical pesticides and this microbial pesticide, there is a very this one process. That's why there is a little bit limitation is there, ma'am.
But can't we go for I mean preparing at our end in our labs and all at a lower price, which is affordable to the farmers?
Yes, ma'am. Yes, ma'am. People are nowadays doing on this. Actually, for this experiment also, ma'am, I have bought from Tamil Nadu, from the Green Life Biotech lab when I was in OUAT Bhubaneswar campus.
Because private people are minting money like anything in agriculture also. When we focus on such aspects which are really useful to the farmer, I think we can convince farmers and they will adopt this on a larger scale.
Yes, ma'am.
I think this is from my end, sir. If any questions are there, they are welcome.
मैडम हर्षदीप, आप पूछना चाहेंगे कुछ? किसी का कोई क्वेश्चन? रोहित जी नेक्स्ट करिए।
जी सर। Gitanjali ma'am, are you there?
Yes, sir.
We can proceed with you, ma'am.
Yes.
Okay, thank you.
Sir, I think I'm not able to share. So can you give me 5 minutes? After another person can.
Madam, the voice is very low. I think it is not audible, please.
गीतांजलि जी आप प्रॉपर नेटवर्क जोन में नहीं है क्या? आवाज नहीं आ रही है आपकी।
So is my screen visible?
नहीं, मुझे तो नहीं है विजिबल।
Sir, I think there is some glitch. I'm not able to share my screen.
No, it will take some time, madam. 1 minute like that, you wait. Are you sharing from mobile?
No, ma'am, I'm sharing from laptop, but I think
laptop only means you just click it and it will take 1 minute for sharing the screen.
Yes, ma'am.
It's okay, ma'am. We will move with Isha, ma'am. Then you will do the presentation. Isha, ma'am, are you there? Hello? Hello?
ईसा जी आप हैं?
I think we can go with the next presenter, sir.
हाँ हाँ यही कर रहे हैं मैडम।
Yeah, I think she should have been able to share. I don't know what is wrong.
और नेक्स्ट में देख लो मानसी शर्मा वगैरह ये सब है, इनका करा दो।
Ripanda Kaur, are you there, ma'am?
Yes, sir.
Okay, we can proceed with you, Ripanda, ma'am.
So is my screen visible?
Yes, ma'am.
Yes, madam. Screen is visible.
Okay, sir. Good evening, everyone. My name is Ripandi Kaur. I have completed my bachelor's in February this year from Punjab Agriculture University, Ludhiana. And I am here to present a poster on the topic Light Emitting Diodes or LEDs as a Sustainable Approach in Post-Harvest Management of Horticultural Produce. As we all know that horticultural commodities are the most perishable among the farm produce, almost 30 to 50% of the produce is deteriorated during post-harvest handling, incurring heavy losses to the farmers. The conventional methods of using chemicals, they are being criticized by the society because of the harmful effects they are posing on the human health and the environment. Therefore, it has become a necessity to shift towards a sustainable approach as it is durable, economical, and most importantly, it is very environmentally friendly. It helps to improve the overall quality of produce, thereby fetching higher price in the market and benefiting the farmer. So how does an LED work? LED, it consists of a semiconductor with a positive and a negative junction. When it is connected to a power supply, flow of current starts from positive to the negative junction, resulting in the flow of electrons, which thereby emits the light of a certain wavelength, which is then used to treat the post-harvest produce. How it helps to improve the quality of the horticultural produces? It helps to reduce the losses by enhancing ripening process and delaying the emergence of ethylene. It also helps to enhance the color of the product. It helps in suppressing the disease occurrence and improve the overall nutritional quality by improving the content of bioactive compounds such as vitamins, minerals, flavonoids, total soluble sugars, etc. Moreover, it also helps to delay the senescence process and extend the photosynthesis period of the plants. Coming to the findings, on illuminating the broccoli with red light at 660 nanometers, delay in senescence and reduce in yellowing and less ethylene production was observed when compared with blue and white LED, and on illuminating with deep UV at 272, 289 and 293 nanometers in strawberry helped to prevent food spoilage by mold growth. To conclude, though there are some limitations of using LEDs as it may cause moisture reduction and reduction in the mass of the produce, but when used properly at a proper light intensity and proper duration, it can give tremendous results because it is environment friendly and a safe approach rather than using any chemicals. The most effective wavelengths for plant photosynthesis are blue and red at 400 to 500 nanometers and 600 to 700 nanometers respectively. Thank you. Hello.
Yeah, madam, these findings are very good, but because ours is a farming community and all, how far do you think that there are efforts required to take these techniques to the farmers?
Yes, ma'am. This is a very under लोगों को इतना पता नहीं है इसके बारे में. So if we will reach out to the farmers, then it will definitely help them. We could develop many extension programs to take this research to the farmers.
Especially because these are all horticulture crops. I think there can be a linkages between the horti wing with this particular firms as well as the extension unit, and maybe even government involvement should also be there in promoting when you have such a good results, especially export oriented vegetables you have focused, isn't it? Otherwise, if these findings don't go to the farmers, it is only theoretically in the books and articles.
Yes, ma'am.
So are there any efforts in your university which are done in this line for popularizing them at the farmer's end?
No, ma'am, not much yet. But when I'll go for my master's, then I'll definitely focus on this.
Yeah, because they should also know that these are the results which could do wonders in their fields. Otherwise, as I have told you, it will be only pertinent to the books and knowledge, that's all.
Yes, ma'am, exactly.
The working principle and all training should be given, and brainstorming sessions, especially to the horticulture farmers and horticulture wing.
Yes, ma'am.
Yes, sir. Still any more queries on this aspect? So we have a lot of demand for the colored capsicums and broccoli nowadays because of the health benefits and nutrient profile.
Yes, ma'am.
Yeah, is
I
it?
think we can next
Yeah.
participant, ma'am. Yeah. So
Yes,
Gitanjali,
please.
are you there, ma'am? Gitanjali, are you there, ma'am?
Gitanjali, again, I think she has a problem. She has given in chat, sir.
Yes, sir. Sir, I tried to fix it, but still it is not sharing. I don't know what is the problem.
So you could log off and try once, madam. Till in the meanwhile, another.
Okay, ma'am. Yes, ma'am.
Another participant, if anyone is there, I think they can go ahead.
Isha ma'am is there, I think.
Yeah. So that after her, she will try to join.
Isha ma'am, are you there? Ma'am, audible to you or not? Yeah, I already make you co-host, ma'am. You can unmute yourself.
Yeah. Once you can log off and log in, madam, that will solve the problem, at least for one of you.
Isha ma'am, Gitanjali ma'am, anyone is there? Ankur sir? Hello, Ankur sir?
हाँ जी बताएं।
सर आई थिंक मैम का नेटवर्क नहीं चल रहा है, दोनों मैम का, तो आई थिंक हम अब आज कंक्लूड कर सकते हैं। अभी कल इनका करवा देंगे सुबह।
आज कंक्लूड 10 बजे से आते हैं।
We'll complete my oral presentation, sir, second one. So in 5 minutes, so that we can start tomorrow with the posters.
Sure, ma'am.
जी जी।
That would also complete everything in the what we say oral.
तो मैम कल 10 बजे स्टार्ट करें, फिर 11 की जगह 10 बजे।
I meant to say is now I will close the session with my oral, one more oral, sir, so that tomorrow.
अच्छा ओके।
हाँ,
हाँ
tomorrow.
फिर स्टार्ट, हाँ ठीक है, पोस्टर से स्टार्ट करेंगे कल।
Today we'll finish off this one, sir. Oral one is left out. No?
प्लीज मैडम आप अपने स्टूडेंट को कराइए।
Start there, sir. I am doing because she is giving the credit seminar there.
Okay, no issue, madam, no issue.
Okay,
Please.
yes.
Okay.
Am I audible and the slides are visible, sir?
Yes, ma'am.
Slide is visible, sir? First
Yes,
slide.
ma'am. Yes, ma'am.
So, good evening once again. So, this is one of the study done by my student on heat and soil physicochemical properties as influenced by conservation agricultural practices in castor-based cropping system under rainfed conditions. So basically, this is the work done on the Indian Institute of Oilseeds Research as a part of the research program, and some part has been given to the student, that is Mr. R. Sai Mindra, now a PhD scholar at Navsari Agriculture University. So I hope the introduction is visible, sir. Is the next slide visible, sir?
Yes, ma'am. Yes, ma'am.
Yeah. So I just don't want to focus much on the written material, but as all of us know, that in the present scenario of the climate change, due to our agricultural operations or the human activities, apart from the severe occurrence of the heat waves, which we are observing now, storms, floods, and droughts, they pose a threat to ecosystem and agricultural production, apart from the well-being of the human beings. So, as a part of this, to avoid all these events and as a mitigation strategy, so conservation agriculture has been a very good sustainable aspect in the agriculture, which is mainly focusing on three principles, that is zero disturbance of the soil, and then the minimum disturbance, what we say, then what we say have the cover crops and diverse spread cropping system. So as a part of this only, the study was also taken for the student in the castor cropping system with the three types of tillage. So when we see the technical details, the location was the one of the research farm of Indian Institute of Oilseeds Research, that is Narkoda farm, near the Rajiv Gandhi International Airport, that is Samshabad, 10 kilometers away from that. And the season was Kharif 2021. Actually, this is the 9th cycle, 8th to 9th cycle, I believe. But we had taken during the 3rd to 4th year cycle. The experiment for 8 to 9 years they have planned. And the experimental design was split plot. And as I've told, it is in a castor-based cropping system as a, what we say, the subplot. The castor with a spacing of 120 by 45 centimeters, which is a long duration as well as a wide row crops. And the three intercrops chosen were three pulses, that is, one is red gram, groundnut, and green gram. Basically, one of the, what we say, the oil seed crop. And the castor variety was ICH 66, and red gram was red gram 76 with 120 days, and green gram, Warangal green gram 40, and groundnut was K6 Kadiri 6, a popular variety, still rolling now. And the recommended dose of fertilizer is 80, 40, and 30 kgs of NPK per castor, and the soil type was alpha soil, that is red soil. And coming to the treatment details, as I have told you, three tillage practices, that is conventional tillage, where one is two cultivator and one rotavator, and reduced tillage, one cultivator and one rotavator. No disc was used, and zero tillage where there was no disturbance. So these were the three tillage operations in the main plot, and subplot it was intercropping systems with castor, that is sole castor, castor and red gram in 1:1 ratio. And here, like the whatever residue was there of the previous crop, it was spread there, and the castor plus green gram, and the previous crop was incorporated here as a mulch, and castor plus groundnut in 1:3 ratio. So when we have a gist of the experimental results, we could see that the main plots and the subplots on the yield attributes and yield of castor. So the conventional tillage, as usual, it has given a higher yield attributes in castor, that is capsules, spikes, and seed and stalk yield. But it was interesting that it was closely followed by the reduced tillage in all the aspects, while the zero tillage has resulted in lower, because it was the initial years, like second or third year, where the experiment was taken and the observations were taken. Coming to the subplots, that is the different cropping systems, the sole castor has recorded higher yield attributes and yield, but it was closely followed by castor plus red gram intercropping system, as well as castor plus groundnut. So these two crops along with the castor had a anidation or complementary effect when compared to that of the what we say, the short duration crop, that is green gram, which has recorded, I can say, lower yields of what we say, the castor. But we can see that like the stalk yield was only significantly superior over here, which has got a little negligible requirement for the agronomy aspects. Yeah, when we see this figure, like what is the castor equivalent yield among the different treatments? Interestingly, we can see here that, as usual, the conventional tillage, it has recorded the higher equivalent yield, but it was closely followed by the reduced tillage and the lower, comparatively lower with the zero tillage. And among the cropping systems, it was castor plus red gram, and then it was castor plus groundnut in terms of the castor equivalent yield. Coming to the soil moisture, this is one of the important aspects. There was a completely rain-fed situation during all the years of the experiment. So we could see that among the tillage practices, invariably the moisture content was higher in reduced tillage as well as the zero tillage, whereas the lowest was seen in conventional tillage in the top 0 to 15 centimeters as well as 15 to 30 centimeters depth. And coming to the intercropping systems, the moisture content, here also we could see the spectacular effect of what we say the castor plus red gram as well as castor plus groundnut in conserving the soil moisture. And then it was sole castor because it was for a longer period and due to the what we say coverage in the soil, whereas the castor plus green gram comparatively lower soil moisture was observed as compared to that of the other intercropping systems. And when we see the most interesting feature in terms of the farming community as well as for the profitability of the system, we can see here that the benefit cost ratio as well as the net returns were higher for the our reduced tillage because lower cost was involved there, although little yield reduction was there as compared to that of the conventional tillage. And coming to the cropping systems, when we see at the benefit cost ratio and net returns, the highest was with castor plus red gram owing to the higher cost of red gram. And we could see that it was castor plus groundnut in the next place. So in all the aspects, as we have seen, castor plus red gram was the dominant one in terms of yield as well as the moisture content as well as the benefit cost ratio. So based on this small researchable objectives, we can conclude that under the tillage systems, the reduced tillage was found equally efficient as that of the conventional yield attributes and yield of castor as well as soil moisture conservation. And one slide, I think I have skipped it. That is, soil organic carbon and what we say the NPK availability. Here also we could see that the organic carbon, there was a slight increase as over the conventional tillage in reduced tillage and zero tillage because the disturbance as well as the addition of the organic matter, whatever was there, is higher in this and being a third cycle of the experiment. And the nitrogen, phosphorus, and potassium buildup was also a little high as compared to that of the conventional tillage. And among the intercropping systems, it was higher with red gram and castor as well as castor plus green gram owing to more foliage and drop comparatively, and almost all it was nearer with the castor plus groundnut over the sole castor because it is only a single crop and the nitrogen accumulation from the legume crops is higher, as it is most clearly evident that all these three were legume crops as compared to that of the sole castor. So based on that, so reduced tillage was equally effective and it was effective in soil moisture conservation as well as the buildup of the NPK and organic carbon in the soil. And the cross returns as well as the BC ratio were also on par with the reduced tillage as compared to that of the conventional tillage. And these positive results could be still understood very well by a long-term research, which could be continued over some more years. And especially we want to recommend that for the semi-arid regions, which are characterized by the drought and uncertain rainfall, the adoption of the cropping sequence or cropping system, intercropping system along with the castor, which is a resilient crop in the semi-arid crops, under the reduced tillage or on a long-term basis, zero tillage is fetching to the farmer, to the soil, to the environment, as well as to the what we say the human needs. So I conclude this with some photographs of our research, that is castor and some other remaining crops. Thank you. So I have come to the end of the presentation, sir. Any queries or doubts?
यदि किसी की कोई क्वेरी या डाउट है तो पूछ सकते हैं आप. मुझे तो नहीं लगता, क्योंकि मैडम ही सबकी डाउट पूछते हैं, तो मैडम से कोई पूछेगा.
Not like that, sir. But the good thing is that it is completely a rain-fed, and I have visited many times the experiment also, which is worthwhile seeing and also results going to the farmer.
बहुत बढ़िया मैडम, बहुत अच्छा, बहुत ही बढ़िया। तो मैम कल का फिर क्या शेड्यूल, 10 बजे से स्टार्ट करें कल से पोस्टर?
Sir, because we can at least start at a correct time.
11 पे ही करें?
10 o'clock would be better, sir. Early is the best. Because at least if we start by 10, 10 मिनट ये यहाँ वहाँ हो जाएगा, 11 में फिर लंच ये सब बोलेंगे, तो at least we can wind up quickly. 10 would be better, sir.
जी ठीक है, तो 10 मैडम स्टार्ट करते हैं। तो विद योर ड्यू परमिशन आपका सेशन फिर एंड किया जाए?
Yes, sir.
जी जी ठीक है, थैंक यू मैडम।
Thank you, sir. Thank you for the opportunity. We'll join by 10, sir.
जी
Tomorrow.
ओके, थैंक यू सो मच मैडम फॉर योर काइंड हेल्प एंड सपोर्ट।
Sir, because it is our duty as a agronomy person.
जी जी जी, थैंक यू मैडम, थैंक यू मैडम, थैंक
With your
Okay.
permission, we will leave, sir.
ओके, थैंक यू मैडम, थैंक यू, ओके प्लीज।
Madam,